you had a very powerful question at the very beginning of your career to where if adult salamanders can regenerate damaged tissue, then why can't humans?
Guess what? Yeah, they can. And the problem we have seen so far is the fact that we've been keeping humans under the wrong environmental conditions for regeneration. So if we change those conditions, we can have regeneration. People think high hyaluronic acid is lubricant, and it's not. High hyaluronic acid functions as a molecular sieve to keep bacteria and viruses from attacking articular cartilage.
That's like a very standard treatment that orthopedics do in an office.
Exactly. People don't understand what they're working with. I have people that say, adult stem cells don't exist. I found them. They do exist. So, I mean, don't believe dogma. Just because everybody else says this is what happens doesn't necessarily mean this is what happens.
When you use the word regeneration in human medicine, like what do you mean by that?
Restoration of the damaged or missing tissues, creating the normal histoarchitecture. If you have that correct, you restore function.
Telomerase positive and then stem cell.
It is what I call the true stem cell. It is pre-programmed to heal. That's its only function. It's not maintenance. It's not function. It's healing, repair, restoration, regeneration.
What is the difference between a telomerase positive versus a telomerase negative cell?
Well, one has unlimited proliferation potential because of the telomerase enzyme. Telomerase negative doesn't have.
I have the pleasure to be here with Dr. Henry E. Young, PhD. And today we're going to be covering some very important topics in stem cells and the regenerative medicine world. And I think you guys are going to have a lot of good information, especially coming from Dr. E. Young, from Henry E. Young. He's been at the forefront, especially when he's been in his research studying adult telomerase positive stem cells. And I have
ATPSCs
today
are easier to pronounce.
ATPSCs, yes. And so what I'm going to do now is I am going to be playing a video. And so before we start the video, so there's two videos that Dr. Young wanted me to show you guys. And in these videos, it explains what ATPSCs are. Each of the videos are about 7 minutes long. And then also from there, we'll start our conversation, the podcast. And then we will also be able to show the video. You guys have access to it. There's also some documents, some flow charts that he presented to me, that he shared with me that we're going to be going over as well. And then you guys will be able to download those. And then the biggest thing too is this is just the first episode. We have, I believe, 7 more episodes that we're going to get more into the details of the ATPSCs and what specialties and, more of his research that we're going to be covering. So I'm excited for this. So let's get into now the video.
Welcome to The Explainer. You've probably heard the hype about stem cells, right? But what if I told you the most powerful ones aren't cooked up in some lab? They're already inside you. Today, we are diving deep into our body's hidden healers, a type of stem cell that could totally revolutionize medicine. Let's get right into it. Okay, so let's kick things off with the big problem in regenerative medicine today. Cells like embryonic and mesenchymal stem cells. For the longest time, they were seen as the holy grail, the future of healing. But as this quote from the research points out, when it came to actual clinical trials, well, their effectiveness just wasn't what everyone hoped for. They kind of fell short of the hype. And here's the puzzle. On one side, you've got embryonic stem cells. These things are incredible. They can turn into any cell type in the body. The problem? Well, it's a big one. They carry a risk of forming tumors, these things called teratomas. Yikes. Then on the other side, you have adult stem cells, like MSCs. They're much safer, for sure, but their success rate in trials, it's often really, really low. We're talking sometimes just 1 to 5%. It's like you have to choose between high risk or low reward. Not great options. So what if we're looking at this all wrong? What if the perfect stem cell isn't something we have to pick and choose the risks for? What if it's been hiding inside our bodies this whole time? And that, my friends, brings us to this incredible, kind of unsung group of healers living in our own tissues, adult telomerase positive stem cells. So, what exactly are these ATPSCs? Well, think of them as this super rare population of stem cells that you're born with, and they stick around for your whole life. They're not engineered in the lab or anything like that. They're a completely natural part of you, and they've got a secret weapon, something that gives them this unbelievable power to heal. And that secret weapon? It's an enzyme called telomerase. Seriously, this one little thing is what makes ATPSCs totally different from pretty much every other adult cell in your body. It is the key to their amazing potential. All right, to really get why telomerase is such a game changer, you have to understand how most cells get old. See, most of our cells have this biological clock. It's called the Hayflick limit. It means they can only divide a certain number of times, maybe 50 to 70, and then that's it. They age and die. But ATPSCs, they play by different rules. Telomerase is like this protective little cap at the end of their chromosomes. Every time a normal cell divides, that cap gets shorter, but telomerase keeps rebuilding it. This means they can keep on dividing, basically forever, as long as they're in that stem cell state. And check this out. This table really breaks it down. ATPSCs have that telomerase goodness, just like embryonic stem cells, but, and this is huge, they don't have that scary tumor risk. Plus, unlike mesenchymal stem cells, which we have fewer and fewer of as we get older, our number of ATPSCs stays the same our whole lives. It's like they have all the pros of the other cells with none of the cons, the best of both worlds. Okay, let's pause on this diagram for a second, because it really shows you just how versatile these cells are. See, it all begins at the very top with what's called a totipotent cell. The name says it all, total potential. It can literally become any tissue, including the placenta. From there, it's a one-way street. They differentiate into pluripotent cells. Now, these are still super powerful. They can become any of the body's tissues. We're talking muscle, nerves, bone, anything. And from that point, they specialize even further into the specific cells our body needs. It's this beautiful, organized waterfall of potential, and it's all happening right inside of us. So, where can you find these powerhouses? The short answer is everywhere. They're hanging out in our connective tissue, the stuff that basically holds us all together. They're in your skin, they're surrounding your organs, they're even wrapped around your nerves. It's like we have this internal repair crew stationed all over the body, ready to go at a moment's notice. Okay, so if these cells are all over the place, just waiting, how do we get them to spring into action when we actually need them? How do we wake up this internal repair crew? Well, in their natural state, ATSCs are basically sleeping. The scientific term is quiescent. Think of them like a team of elite paramedics, just on standby, waiting for the 911 call. They don't do anything until the body sends out a distress signal, a sign of injury, damage, or some kind of chronic disease. Now, one way to get them working is pretty high tech. It involves taking a tissue sample from the body, using some really complex lab techniques to find and separate these super rare cells, growing a whole bunch of them in a dish, and then putting them back into the body. It definitely works, but it's, a very involved and technical process. But that leads to a really cool question, right? What if there's an easier way, a more direct way to wake up these little healers without having to go through all that lab work and taking them out of the body? And unbelievably, the research points to a much simpler answer, a special supplement. It's called a combinatorial nutraceutical supplement pill, or CNSP. The whole idea here is to use a specific mix of natural ingredients to wake up the body's own APSCs right where they are. They call it in situ activation. Basically, you'd be mobilizing your own internal repair crew with a pill. It completely flips the script on medicine, from an outside-in approach to an inside-out one. Okay, this all sounds amazing in theory, but what about in practice? Does this potential actually lead to safe and effective treatments for real people? Let's take a look at the evidence. First off, safety. In the clinical studies where they isolated these ATPSCs and put them back in, the safety record has been perfect, 100%. Across multiple official studies, there wasn't a single reported adverse effect. That's a huge deal. And it's not just safe, it's effective. We're talking a remarkable 86.4% success rate in actually reversing the symptoms of disease. And get this, that's not just for one or two things. That number is a cumulative result across 20 different chronic diseases and major injuries. And the list of conditions they've studied is just staggering. We're not talking about minor aches and pains here. We're talking about some of the toughest diseases out there. Neurodegenerative stuff like Parkinson's and Alzheimer's, but also things like osteoarthritis, damage from heart attacks, COPD, and even traumatic spinal cord injuries. The potential here is just wow, it's huge. So let's just take a step back for a second and think about what this all really means. This isn't just a small step forward. This research could fundamentally change how we think about healing and regeneration. The biggest takeaway here
Mhm.
is that the future of medicine might not be about creating some new wonder drug that we put into our bodies. It might be about figuring out how to unlock the incredible healing systems we already have. It's like each of us has this powerful, untapped pharmacy inside, and we're just now finding the key. And that leaves us with one last kind of mind blowing thought. If our bodies really do hold their own blueprint for repair, a system that doesn't really age and can become any tissue we need, then what's the limit? What aren't we capable of healing?
Amazing. Okay. So this is the first video. That's what I watch. I watch a whole bunch of house music, so I apologize about that. This is now the video number 2. And I know you said that you wanted to give credit to Thanos for making these videos, Dr. Young.
Yes, I do.
Okay. Yeah, I forgot. I know we were talking about it off camera and didn't bring that up. Okay. So this is the second video, second part.
For decades, we've been looking for the next big thing in medicine, right? Some holy grail of healing. But what if I told you the most powerful repair kit imaginable wasn't in some high-tech lab, but was already hidden inside every single one of us? Today, we're diving into the incredible discovery of Dr. Henry E. Young, a built-in healing system that could literally change everything we thought we knew about medicine. But let's start with a question we all ask ourselves sooner or later. Why do our bodies wear down? Why do we get arthritis, heart disease, all these things that just seem like an inevitable part of getting older? Is it just a flaw in our design, or is there something bigger going on? Well, the conventional answer has always pointed to our cells. And the problem, as science has seen it for a long time, is that our bodies are kind of designed with a built in finite clock. So here's the deal. At the very heart of this is a kind of cellular timer. Most of the cells that make up our body, our tissues, our organs, are what you call telomerase negative. And what that means is they have a hard limit on how many times they can divide. For us humans, it's about 70 times. After that, they get old, they stop working right, and eventually they die. It's like a pre-programmed biological clock, just ticking away. And this clock ticking down isn't just some abstract concept. It has very real, very painful consequences. I mean, think about osteoarthritis. That's the slow, grinding breakdown of our joints over decades, all because the cells just can't keep up with repairs anymore. Or think about what happens after a heart attack. The damaged muscle can't regenerate, so it gets replaced by scar tissue that can't pump anything. That is our biological clock in action, and it leads directly to these degenerative diseases. And the scale of this problem, it is absolutely staggering. Just cardiovascular diseases alone are responsible for 30% of all deaths worldwide. That's our biological clock at work on a massive global scale. Okay, so that sounds pretty bleak, right? But what if that whole idea of a finite clock wasn't the whole story? What if our bodies had some kind of an override switch? Well, this is where a truly revolutionary discovery changes the game, revealing a hidden repair system. Back in 1975, Dr. Henry E. Young found something incredible. A very, very rare population of cells scattered throughout our body's connective tissues. He named them adult telomerase positive stem cells, or ATPSCs for short. And these are not just any old cells. They are basically the body's own master healing cells. So, what makes these particular cells so incredibly special? What's their secret? Well, it turns out they're playing by a completely different set of rules than almost every other cell in your body. In a very real sense, they are biologically immortal. The secret sauce, the big difference maker, is an enzyme called telomerase. Now, remember how we said most of our cells are telomerase negative with that ticking clock? Well, APSCs are telomerase positive. This enzyme acts like a little maintenance crew for our DNA, allowing these stem cells to divide over and over and over again without ever getting old. They basically have unlimited potential to create new, healthy cells. And get this, it's not just one type of cell. Dr. Young actually figured out there are 8 distinct subcategories. They range from the incredibly tiny but all-powerful totipotent stem cells, which can become literally any cell in the body, all the way to more specialized cells that are already programmed to become things like muscle or bone or skin. It's like having a full construction crew on standby. ready to rebuild anything that breaks. This chart really lays out the whole process perfectly. You can see it's a hierarchy, right? It starts with those most powerful, most primitive cells at the top. When they get called into action to, say, repair a muscle, they start to differentiate. But here's the crucial trade-off. As they specialize and go down this one-way path, they lose that telomerase enzyme. So the brand new cells they create are born with that finite 70-division clock, while the original stem cell reservoir stays immortal, ready for the next call. Okay, so the science is just mind-blowing. But what does it actually mean for treating diseases? This is where we get to move out of the lab and into the real world to see how we can literally rebuild the body from the inside. Okay, check this out. Let's look at this amazing study on osteoarthritis. Researchers took these activated ATPSCs and used them to treat severely arthritic joints. But the subjects weren't just people. No, they included a German shepherd, a spectacled bear, and yes, even a Komodo dragon. This proves that we're talking about a fundamental biological system that works across species. And the results? They were nothing short of remarkable. In just 1 month, every single subject, from the bear to the humans, showed significantly less pain and could move their damaged joints much more freely. The body's own repair kit was actually working. The results in heart disease are, if anything, even more stunning. You had patients starting with a cardiac output that was dangerously low. We're talking 25% or even less. After treatment with their own ATPSCs, look at that jump. Their heart function increased by 20 to 45 percentage points. And these incredible results have been shown to last for over 12 years. Amazing. So, you're probably wondering, how do we actually tap into this incredible power? Well, researchers have developed 2 really distinct ways to activate this internal repair system. 2 paths to healing. The first method is a clinical procedure called ex vivo activation. Ex vivo just means outside the body. It's pretty straightforward. First, the patient takes some specialized supplements, nutraceuticals, to get their body to make more of these ATPSCs. Then, another compound pushes those new cells into the bloodstream. From there, it's just a simple blood draw to collect them. They're activated in a lab, and finally, they're injected right back into the body where they're needed most, like a damaged knee or a failing heart. But a clinical procedure like that isn't for everyone. So a second, and maybe even more accessible approach was developed. It's called in situ activation, which just means in the original place. This method uses a powerful supplement pill, the combinatorial nutraceutical supplement pill, or CNSP. You just take a pill, and it activates your body's own ATPSCs internally, prompting them to go find and repair damage all on their own. No clinic, no injections needed. And if you want to know just how powerful this in situ approach can be, listen to this story. There was a patient with severe heart failure, cardiac output below 10%. They were way too fragile to even consider the ex vivo procedure. But after just 6 months of taking the CNSP pill, their cardiac output shot up by more than 25%. The body just healed itself from the inside out. This all brings us to a really powerful way to think about Dr. Young's work. It suggests that this holy grail we've all been searching for, it's not something we need to invent in a lab. It's this ancient, powerful telomerase-driven repair system that's been waiting inside us this whole time. And that really leaves us with a final provocative thought to chew on. As we search for more and more complex, high-tech cures, what if the most profound medicine of the future isn't something we create at all, but something we simply learn to unlock? The incredible potential that's already waiting inside our very own cells.
Awesome. Okay. Well, hope you guys found some very interesting takes on that. When I first saw it, when you first sent it to me, I was like, whoa. I was like, I've never heard of that. I was kind of impressed and just a lot of questions. So that's why I reached out and I said, I'm like, all right, Dr. Young, I want to talk to you and see and learn more. Okay. So I have so many questions, right? And I would like to first, before we get into the ATPSCs and all that, I would like to talk about how you got here, right? So how you got there. So I know that you had a very powerful question at the very beginning of your career to where if adult, salamanders can regenerate damaged tissue, then why can't humans, right? So I know that was on your LinkedIn and I asked you that question. That was one of the main questions I wanted to ask you. So yeah, can you take us back to like what made you question that and then how did you get to where you're at now?
Fine. Actually, the initial question for my master's thesis was, if aquatic salamanders can regenerate a limb, why can't adult salamanders of the same species regenerate a limb? That's the whole point.
So you said
I had read
aquatic salamanders. I'm sorry to cut you off. So you said aquatic salamanders. There's different. I'm not really familiar with salamanders.
Yeah.
There's different types of salamanders.
They're ocellatos, which are mature juvenile, mature aquatic salamanders. Then you have juvenile salamanders that exist in an aquatic environment, but when they metamorphose to the adults, they become terrestrial. They live in a terrestrial environment rather than an aquatic environment. So that's what I used for my test subjects, were terrestrial adult salamanders. But literature says, well, this is how you keep them in the lab for regeneration. And they were using aquatics. So it was 4 degrees centigrade. They fed their aquatic salamanders beef liver, dangling up during the day, this sort of thing. I tried to do that with the aquatics, with the terrestrial salamanders. Lo and behold, they all died. I went to my chairman and I said, I can't do my research because all my animals died. And he said, well, what do you know about your model system? And I said, they're adult salamanders. He said, with a twinkle in his eye, I might add, he said, you need to dig deeper and find out everything you can about your model system. So went back, did field work with the adult salamanders, found out they lived in a terrestrial environment in burrows in the ground. They only came out at night to feed. Their preferential food source were things that moved, like cockroaches. That's what they love. Although they will eat night crawlers, that's their second preferred food source, and that's the one I used for the laboratory. They spawn like salmon. And I was at University of Arkansas in Fayetteville, which is up in the hills in the Ozarks. And there's a road that went through there, and it had ponds on both sides of the road. And these terrestrial salamanders would spawn, they would breed in these ponds, but it was crazy because they'd they'd live on one side of the road in their terrestrial environment, cross the road, spawn to the pond where they were originally born, breed, and then cross the road back again to get back to their home environment. So my early dates with my future wife were to collect salamanders as they spawned. And they would spawn across this highway. So I would park my car in the middle of the highway, put the hood up, and be looking like I was fixing the car, and all these semis would be back behind me. And I sent my future wife out to collect the salamanders as they crossed the road. Then when I had about 10 or 15 trucks behind me, somebody would walk up and say, can I help you? Is there anything? Well, no, not exactly. And they'd slam my hood down, and all the other trucks would go around me, and then everybody's going, I'd raise the hood again and get in there. And we'd repeat that 7 or 8 times. So we got a good collection of salamanders that way. And that's how I got my initial population, first and second population.
Wow.
Yeah. Things you do to do your research.
That's a great story, by the way. That's an amazing story. Yeah.
That's why I tell people those were our initial dates when we were dating and stuff like that. Of course, from her side, she worked with frogs, and she was interested in the metamorphosis from tadpoles to adults. So we would go hunt for tadpoles, and she just didn't want any little bitty tadpole. They had to be the bufo tadpoles, which the whole tadpole would fit in the palm of your hand, that sort of thing. So I'd be out there with a seine net, usually in the middle of the pond, and being the chivalrous person that I was, she would take the bag, and we'd seine the pond for these bufo salamanders. And one time, I know I'm digressing, but whatever.
No. This is great.
I'm sure the audience will get a kick out of this. Two little anecdotes here. The first one was I was seining in the middle of the pond, so the water was up to my chest. And all of a sudden, I heard a like that. And I looked around, and there was a feather came falling down. And the waves on the pond were like this, and I was going, looked around, what in the world is going on? And then about, I would say 20 feet away from me, this thing popped up. Eyes were about, I'd say at least a foot apart from each other. I'm going, maybe I don't want to be in this pond, this sort of thing. So I slowly got out, and then another bird flew by, and this thing popped out of the water, and a long tongue shot out, grabbed the bird, pulled it back in, and it went underneath the water. So I definitely got out of that pond. But, we were thinking about, bufos will grow to big sizes, and
Wow.
whatever. And then the second anecdotal studies where we were, and this is more on my wife's side than, whatever. We were at a pond, and she was collecting tadpoles, and a snake popped out, looked up at the pond, and looked directly at her. And she looked at the snake, and both were like frozen for like a few seconds, and then they ran towards each other. The snake went right between her legs, and he just kept hauling ass the other direction. And I was sitting on the bank just laughing my head off of the reaction of what was happening for the tadpoles. So anyway, I digress. Anyway, with the rest of my studies, I found out that during the first cold rain of the fall is when these adult salamanders spawn, and they don't eat during their spawning, and they don't eat when they breed. They only have one thing on their mind, and it isn't eating. It's copulating with their opposite gender. Once they're done, then they return to their burrows. When I presented my findings to my chairman, he told me 4 things that I need to follow throughout my career. And that was, you have to know your model system. You have to know what you're working with. 2, tissue never lies. People may lie to you, but tissues will always tell you the truth. 3, you need to understand what your model system is telling you, and then you have to act accordingly. And the last one is, just because something hasn't been reported does not mean it does not exist. All it means is it hasn't been discovered yet. So, you have people that say, adult stem cells don't exist. Why? Nobody's ever been able to report them, but that doesn't mean they don't exist. I found them. They do exist. So, don't believe dogma. Just because everybody else says this is what happens doesn't necessarily mean this is what happens. You really have to go into the itsy bitsy detail. So anyway, I repeated the experiment with what I would call fat, sassy, and happy salamanders, and I have a picture in my office of one of them. He used to stick out his tongue at me every time I went into the laboratory.
Okay.
Let me get the picture out.
Yeah.
And show you. So, sorry for the break, Annie.
No.
Here's the picture.
Bring it up a little bit. Okay, up a little. There you go.
Yeah, there's a picture.
I
said, he'd stick his tongue out at me every time I came into the laboratory. So anyway.
So is that a natural picture of him? Or you just illustrating
that is a picture of him. That is a picture of him sticking his tongue out at me. Yeah.
That's awesome. Okay.
So then I repeated the experiments. Kept them in their environment, which was terrestrial, burrows, fed them night crawlers. Night crawlers went into burrows at night. Everybody would come up. I had them under red lights, and I could see them go, after the salamanders would go after the worms, and then gobble them up and look around for another worm and then go after it. It's really cool and neat. I should have videotaped it or something. But anyway, because the aquatic salamanders were viewed every 5 days, and it took them anywhere from 30 to 45 days to regenerate, they looked at them every 5 days, and then concluded that adult tissues de-differentiated into the blastema, which then regenerated the tissue. The adult salamanders took well over a year to regenerate, but I kept the observation times at every 5 days. So instead of 6 to 9 observation time points, I was 74 to 100 observation time points, which means I could really dissect what was going on in the tissue. And what I discovered were some very interesting points. After amputation of the limb, a transitional scar forms, covering the wound site. It's basically a Band-Aid. separating a very hostile external environment from a very delicate internal environment. Next was the appearance of macrophages that appeared underneath the transitional scar and basically cleaned up all the debris, dead, dying cells, bacteria, viruses, fungi, mold, whatever was there, cleaned it and made it a sterile environment. Then there was the formation of what I call the apical epidermal ridge. It's equivalent to the apical epidermal cap in aquatic salamanders. And it basically increased in size and then started secreting glycoproteins, specifically sulfated, carboxylated, and neutral, into the underlying tissue. After that, one could see the very small cells covered in heparan sulfate proteoglycan that broke away from the non-transected, more proximal tissues and made a beeline straight for the blastema along that concentration gradient of glycoproteins. There they started proliferating and formed a mass of cells. And then as that mass proliferated, pushed the AER away, you formed a concentration gradient of differentiated cells. Fully differentiated where the proximal tissues were, undifferentiated where the AER was, and everything in between, everything in between. And as the length of the appendage, more proximal intermediaries turn into adult differentiation. So it was a progressive differentiation as the limb outgrew or grew out, as the case may be. Bottom line in the experiments, adult salamanders regenerate perfectly fine. Not a problem. So that led to the question that you asked, that I asked also, if adult salamanders can regenerate, given the correct environmental conditions and the correct nutrition, yada, 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 can adults do this? Can humans do the same thing? Guess what?
We can?
Yeah, they can. And the problem we have seen so far is the fact that we've been keeping humans under the wrong environmental conditions for regeneration. So if we change those conditions, we can have regeneration.
And I'm sure that environment, I mean, there's so many different factors, from external factors to internal, I mean, from what we eat.
I mean, there's nutrition, there's building blocks, there's exercise, there's sleep, there's meditation.
External factors that happens, whatever we breathe, that we cannot control.
Exactly. Now, you asked me the question, and I'll ask it here. Why did you ask that question, and why did it become important for your career? I mean, that's the question you have on my list. Multiple members of my family, my parents, and their progeny have had, as the case may be, serious medical conditions, both acquired and genetic. If these previously unrecognized cells were present in humans, could I actually restore the health of the individuals in my family? And that was my whole goal. And it started my 50-year quest at doing this. So you can come up with your next question. I have it on my list too, but.
Yeah. Okay. And so why do salamanders matter to you? Obviously, like, we understand, and that asks you a bigger question. So, like, what other questions can scientists ask themselves about healing and regeneration?
Okay, adult terrestrial salamanders are the highest order of a mammal that will completely regenerate a limb, giving an exact duplicate of what was lost. Now, scientists can take that and look at the genetics of it. They can look at genetic control. They can look at the extracellular matrix involved. They can look at the glycoproteins involved. They can look at biological active factors involved. The time sequence of how it occurs, what needs to be shown, what needs to show up when to have it occur. If it doesn't show up, what happens, and on and on and on. I mean, you can equate what happens in adult terrestrials to what happens in humans. You just have to figure it out. That's all. So it opens up a whole plethora of things scientists can study.
Yeah. Yeah. Well, that's the biggest thing, right? Figuring out why, right? Or how, right?
Exactly.
That's what your quest is all about, right?
Yes, yes.
When people think about regeneration, okay, they think about regrowing, and I know also the FDA, when we're in a very regulated field, right? With the things that we can say and things like that. When you use the word regeneration in human medicine, like what do you mean by that? Like, are we talking about like tissue repair, tissue
Okay.
replacement? What exactly do you mean?
Okay. What I mean is restoration of the damaged or missing tissues, creating the normal histoarchitecture. I mean, each organ and tissue body has its own fingerprint of the different cell types that make it what it is. That's the histoarchitecture. If you have that correct, you restore function. So that's what I consider regeneration. Restoration of histoarchitecture, return to function, normal function.
So not like adult territorial salamanders where they regrow a limb. Is that considered
No,
regeneration
I
too?
mean, they are restoring missing tissues. That's the whole
That's
point.
that is
And
true.
they're
That's
doing
true.
it to the correct histoarchitecture. You have the skin in the right place. You have the dermis in the right place. You have the muscles extending, skeletal muscles extending. They surround periosteum, which surrounds the bones. There's nervous tissue present. There's vasculature in the correct configuration for the limb.
Yeah.
That's what I call the histoarchitecture.
That it just takes it takes a lot for me to really understand that we can do that, right? That's why I'm like, what do you mean by restoring? But
Yeah.
you are
Okay.
absolutely
Now another thing is, for an anatomist, you have skeletal muscle of the organ and skeletal muscle of the tissue, or for a biochemist or anything like that. For anatomists, they go to skeletal muscle of the tissue. And the reason I say that is I postdoced for a biochemist. I wanted to learn how to isolate extracellular matrix components because at the time they thought those were the ones that directed this procedure. So I said, you have skeletal muscle. They said, yes. He said, okay, what are the components of skeletal muscle? He said, skeletal muscle. Muscle is muscle. I said, not exactly. I said, you have myotubes. They're surrounded by an endomysium. They're surrounded by a plasma membrane. Then they have a basement membrane. Then they have an endomysium. In the mysium are capillaries, lymphatics, and ATPSCs. They are collected into a bundle called fascicle that's surrounded by perimysium. They have arterioles, venules, lymphatics, nerve fibers, and they also have stem cells. Then you have multiple fascicles in a bundle, entire bundle, which is actually the muscle itself. It's surrounded by an epimysium, which has the muscular arteries and veins penetrating to do, on and on and on. And I said, that's what skeletal muscle is. He said, no, it's muscle. And I, because he isolated proteoglycans from muscle. Actually, one of his people did, David Carino, and they took entire muscle, ground it up, and then extracted it and did the isolation. And I'm saying, what are you isolating? I mean, you got fibroblasts in there. You got so many different cell types. How do you know what's doing what? And he said, it's skeletal muscle. And I just, walked out the room, this sort of thing. For 4 years, I made that. We had this back and forth about what a biochemist sees when they look at tissue and what an anatomist sees when they look at tissue. And it's juxtaposed. I mean, so it's like grind and fine for a biochemist and slice and dice for an anatomist.
Yeah.
Whatever. Okay.
what has kept you focused after studying for, you said, 50 plus years, right? And you still continue, what has kept you focused in just
my
studying
family members are what keep my focus. I started the characterization, took me about 30 years. Then I started doing preclinical animal studies. And then we got to, and during the preclinical animal during the characterization studies, my father passed away. So that, lost him. During the preclinical animal studies, I lost my mother. But I still had the progeny in my family that were still, genetics were as it is or was, whatever. My father had heart problems and he was diabetic. My mother had autoimmune diseases. She had pulmonary fibrosis and she had dementia. We don't know about the dementia, but the progeny inherited everything from both parents. So they have heart problems, they have diabetes, they have autoimmune problems, they have pulmonary fibrosis, and a whole slew of other things wrong. They're still there. I'll leave the rest of it to later, but so I have one left to try in fact, they're in the clinical trials right now that we started. So I'll just leave it at that for
So your family, I mean, that, family, people you love will definitely keep you focused and to continue doing what you love or what you think is right or how to help them, right? Okay.
Exactly.
Yeah, I agree. So in medicine, and at least from my knowledge, there's usually three main categories of stem cells that people talk about, right? The embryonic stem cells, the pluripotent stem cells, and then the MSCs, which is the mesenchymal stem cells. I think there's something on the background that whenever you move, I think something. Yep.
Sorry, I'm just, my
No, that's okay.
you're skipping ahead. That's all. You're not following notes. That's okay. That's okay.
No, we cover some of those, so I just want to make sure that we cover the next ones. But the three main categories, right? So embryonic, induced pluripotent stem cells, and obviously mesenchymal stem cells.
Yes.
Why do you think we only hear of those three mainly and not ATPSCs? And I'm sure there's probably other stem cells that we're probably not aware of.
Well, I'm sure there are. I mean, I'm sure you've heard of Muse, MUSC.
Yes.
MUSC cells, Muse cells. There's Miami's, there's MAPCs, there's VSELs. I mean, they're out there in the literature as well. And ATPSCs are out there too. One, it's politics. And two, it's where the articles are published. Okay, politics-wise, before 1990, adults, any type of stem cell, other than bone marrow for bone marrow transplants, which occurred during the 1950s, no other stem cell was known to exist. Then James Thompson in 1990 in Science reported the creation of embryonic stem cells from the inner cell mass of embryos that could do anything. So his original idea was to figure out how inborn errors of metabolism, genetic diseases, spina bifida, cleft lip, cleft palate, things along those lines, leg calprothes, equinus, could be corrected in utero before the baby was ever born. That was the original concept of using embryonic stem cells because there was a moratorium against research on developing embryos and fetuses. So we had a stem cell we had a population of cells that did the developmental process, but we could actually take the cells out, look at the genes, all this kind of thing, and develop techniques that we could treat the baby in utero or the fetus in utero before it was ever born.
Yeah, that makes sense.
Yeah. Then someone had the bright idea that they could use the same cells on adults. And then you started a firestorm. You had the Vatican, you had the right-wing people, Christian, born again, whatever, that said, no, my God, you are killing potential babies to treat somebody else. That's morally and ethically wrong. I agree, it is, to treat adults. Then in 1992, Arnold Kaplan published an article in Science saying that, oh, I've discovered mesenchymal stem cells in adult bone marrow. They'll form 3 cell types: cartilage, fat, and bone. Unfortunately, they decrease in number with increasing age of the individual. So they have a finite biological clock. Then there was the back and forth. Who's better, embryonic stem cells or mesenchymal stem cells? So that was all the rage. And then when mesenchymal stem cells were published, then you started seeing publications on hematopoietic stem cells, on neural stem cells, on liver stem cells, on you name it, stem cells. The problem was these weren't stem cells. They were progenitor cells. Now, you have 3 categories of cells in the body. You have differentiated cells, which are the functional cells. They're composed of parenchyma, which is actually the functional part, and stroma, which is the connective tissue framework. That's category number 1. Category number 2 are progenitor cells. Their function is to replace differentiated cells when they wear out. So, you have a skeletal muscle, it, contracts a lot. It's a multinucleated cell. When it kind of disappears, gets worn out, new myoblasts in this case, which are progenitor cells, add their nuclei, and you form new muscle forming, and it continues to function. But their function is to replace worn out cells. And they do it on a continual basis. And then you have unipotent progenitor cells. Anything that ends with a blast as the suffix is a unipotent progenitor cell, like an osteoblast, chondroblast, adipoblast, fibroblast, anything blast. You have bipotent progenitor cells that will form 2 different cell types. You have a tripotent progenitor cell, like the mesenchymal stem cell. It'll form 3 different cell types: fat, cartilage, and bone. You have multipotent stem cells, like hematopoietic stem cells. It'll form, I think it's up to now 20 different cell types, but they're all within the hematopoietic lineage. They don't go outside their lineage. Then you have what I call the 3rd category, the true, I might add, differentiated cells and progenitor cells are telomerase negative. They conform to Hayflick's limit. They have 70 population doublings before they senesce and die. And they decrease in numbers with increasing age of the individual. Okay, now the 3rd category, true stem cells. They are telomerase positive. They have unlimited proliferation potential. They are present throughout the entire lifespan of the individual. They don't decrease in number. I mean, we've isolated ATPSCs from a 96-year-old that was still as competent as someone from a 10-year-old. Not a problem. So I'm curious about biobanking, but that's beside the point. And they will form, there are 8 categories of ATPSCs, not just 1. You have the totipotent stem cells, by name. They'll form any somatic cell in the body. They'll form gender-specific gametes. So a male will form spermatogonia, but won't form oogonia, and a female TSC will form oogonia, but not spermatogonia, if you get my drift here. They're gender-specific.
Yeah. Okay.
They'll form the nucleus pulposus of the intervertebral disc, which is the only functional adult derivative of a notochord. And the notochord is the primary inducer of the embryo. So it comes off early during embryogenesis. It actually comes off at the 16th cell stage. And the gametes come off at the gametes are the 15th blastomere, and the notochord are the 16th blastomere. And then you have development of the trophoblast, and on for the embryogenesis. Okay, let's see, where are we? Okay, and they'll also form the extraembryonic membranes, placenta and umbilical cord. So A totipotent will do all that, and the ATPC.
Would you want me to pull up that flow chart that you created? Is that accurate, or
Yeah,
you're
sure.
still
It's the top-down flow chart.
the top down?
Yeah, the
Okay.
top-down.
The induction of ATPSCs? Yeah. Okay. You remember I have those handy, so whenever you're referring, because I kind of figured. Let me share it right now.
Okay, yeah, there we go. So totipotents will form, we've been able to show notochord, nucleus pulposus, gametes, spermatogonia. They'll also form placenta. The way we did that was to take explants of those tissues, take the conditioned media and apply it to the cells. And we got those cell types. And we did cross species. We use cross species cells to generate the conditioned media and then apply it on a different species. And then we use species-specific antibodies to detect phenotypic expression markers. So we weren't crossing cell types and this sort of thing. So anyway, the totipotent stem cells will do those. The next one are the halo-like stem cells. And the halos and coronas are transitional stem cells between the toadies and the pleuries, basically is what they are. So, and they are pluripotent. Corona-like stem cells are pluripotent. Pluripotent stem cells are pluripotent. And the germ layer lineage stem cells are pluripotent. And I'm sorry, some of these names are confusing, but that's how it was. So I try to keep convention. Then you have the germ layer lineage stem cells, which differentiate into ectodermal stem cells, mesodermal stem cells, and endodermal stem cells. And they only form cells within those respective germ layer lineages. They lose the telomerase enzyme. They now become progenitor cells. They form ectodermal progenitor cells that are multipotent, tripotent, bipotent, unipotent. And we've been able to show 15 differentiated cell types using the ELICA. We have the mesodermal progenitor cells, and they do the same thing, and we've been able to show 37 objectively identifiable cell types, again, using the ELICA. And then the endodermal progenitor cells, we've been able to induce 14 different cell types. And these were objective assays using the ELICA. We coupled the ELICA with our library of recombinant proteins, morphogenetic proteins, and cell-specific exosomes to derive these different cell types. So if you basically bring up the chart that shows the different bioactive factors, and there's a list, and there we go, okay. We've identified 4 different biological activities. We have inhibitory factors. Leukemia inhibitory factor is one that prevents embryonic stem cells from differentiating. We have anti-differentiation factor, which inhibits. We tested these all on our cells, and it shows the different activities. Caffeine inhibits differentiation. SIF is very specific inhibitor for scarf tissue formation. Then we have factors that induce proliferation, and they're the platelet-derived growth factors, AA, BB, and AB. Then we have progression agents, IGF-1, IGF-2, and insulin, and they take a progenitor cell and force it into a differentiated cell type, or prod it along, if you want to call it that. They have absolutely no effect on the adult telomerase positive stem cells. Then if you go down further on the list, that starts then the different inductive factors we tested. Bone morphogenetic protein 2, the ones we isolated except for BMP2, we isolated the other morphogenetic proteins from demineralized bone matrix, and we discovered that bone matrix is a repository for excess exosomes containing the inductive factors. So if the body doesn't need them, it sticks them in bone, and then when it needs them, it degrades the bone, brings them out, and uses them. Interesting process.
Yeah.
The recombinant proteins, I just developed the chicken ELICA assay, And it was at the time that the genome had just been finalized, and everybody was taking little pieces of it and trying to figure out what those pieces did. So they sent them to my laboratory. And my payment was, okay, I want 5 milligrams of your unknown compound. I will test it. I will tell you exactly what it does, and I get to keep the rest. They agreed. Not a problem. They didn't have to pay me any money. They just gave me what this sort of thing. So I used nanogram quantities of compounds to tell them exactly what it did. And then I got to keep the rest. So that was my library of recombinant proteins. We isolated morphogenetic proteins from bone, and then I did the exosome-specific conditioned media, and we could actually isolate the exosomes too if we wanted to. It was a little more difficult, and conditioned media worked fine. So, and we tested those on different cell types. I did it for my ATPSCs, and I also did it for MSCs, because at the time people were saying, well, your cells are exactly the same. I was saying, no, you're wrong. They're not. They have different functions. They do different things. MSCs, in my hands, would only form, yes, they form cartilage, fat, and bone, but the original person that did the analysis was a biochemist. Okay. Fat, there are two kinds of fat. There's white fat and there's brown fat, unilocular white fat and multilocular brown fat. MSCs only formed unilocular white fat. They form cartilage. Well, yeah, sure, but there are 5 different types of cartilage. There's growth plate cartilage, there is fibrocartilage, there is hyaline cartilage, there's elastic cartilage, and there's articular cartilage.
Yeah.
Each are located in different places. Growth plate cartilage is endochondral ossification. Fibrocartilage is menisci, labrum, symphysis pubis, annulus fibrosus, that's all fibrocartilage. Hyaline cartilage connects the ribs to the sternum. So it's a stiff but flexible attachment. So attachments where you want a strong attachment but flexible is hyaline cartilage. Elastic cartilage is found in the pinna of the ear, eustachian tube, and epiglottis. It's more flexible because it has elastic fibers in it. And then you have articular cartilage that surrounds the surface of bones that are articulating, ambulatory bones. And it has 5 different zones. It has a tangential zone, which is predominantly, and I can give you the specifics, and I think I will. The tangential zone is predominantly keratan sulfate proteoglycan. So that means it's a high number of sulfates. It binds to a sulfated glycoprotein called lubricin, which sets up a dipole with water, and actually the two surfaces glide past each other because of lubricin, not because of high hyaluronic acid. People think high hyaluronic acid is lubricant, and it's not. High hyaluronic acid functions as a molecular sieve to keep bacteria and viruses from attacking the articular cartilage. That's its function. But because
so it's not to lubricate?
No, it's not. Yet people inject high hyaluronic acid into joints to lubricate. It's
like
viscous.
that's like a very standard treatment that orthopedics do in an office, and steroid shots.
I know, exactly. And all high hyaluronic does in the very large form is to prevent bacteria and viruses from attacking the articular cartilage. That's its function.
Yeah, wow. That's interesting.
Yeah, people don't understand what they're working with, guys. I'm sorry,
I as I'm talking to other scientists and yourself, I've realized that taking what you guys find in a lab, like of what the true science is, to then putting it out to practice, to the market, to an actual use for doctors to use, that translation, it's missed on a lot of, like, in every part of medicine, I would say.
Well, one of the problems is, and I'm different because I've been in this thing for over 50 years, is you have somebody isolate and find something and report it, and then drop out and somebody else picks it up and does something with it, drops it out. Somebody else picks it up, drops it, and on and on and on. So you have different people with different ideas about how things work, and there's no continuity in their research. You have a dinosaur like myself, I've got continuity through over 50 years now. I know how things happen. I know what I'm looking for. I know what the normal histology should look like. I can understand the pathology of what it doesn't look like. Like, for example, we did a collaborator and myself did a study, well, a collaborator group and myself did a study on our mesodermal stem cells at the time and how they would form articular cartilage. So what they did was to take the tibia and bore a hole in it, get a biomatrix that would release at a defined rate, stick the mesodermal stem cells, pack it into the tibia, and then see what happens. And they did different biological matrices. They did collagen by itself. They did polycarboxyphenoxypropane, which actually worked the best because it had a nice control rate of delivery and so on and so forth. And what they found out was that, and they used, like marrow cells and whatnot. And what they found was that the mesodermal stem cells formed articular cartilage. Small, granted, but still formed it, where the merostroma cells formed fibrocartilage, or some facsimile thereof. And that was published in our 2004 paper on Cell Biochem Biophys, volume 40, pages 1 to 80. The article took up the entire journal issue. And it had that study in it along with a few other preclinical animal studies we did. So, I mean, again, it depends on which cell type you're working with and what happens, what that cell type will form. We were expecting something, and as it turned out, it formed articular cartilage. And we could see the zones of articular cartilage. And that, I think, also is in that osteoarthritis paper that they referred to in one of the two introduction thingies, and also we published in 2020.
I'll have to take a look at that paper if it's especially about
Well, I
cartilage.
can send you the PDFs. That's not a problem.
Yeah, please. I would like to see that to learn the real
I
science.
have 109. I actually have 109 papers. Would you like to see all of them?
In phases, in batches. In batches, I would.
In batches, okay. Anybody that's interested, I can posted my publication list on LinkedIn several times. And they said, well, I don't want to delve through this. Can you tell me which ones were preclinical, which ones were clinical, which ones were this? So I color coded it.
Yeah.
Here's the color code. Tell me which papers you want, and I'll send them to you. And I'll do that too. Just send me your email address. So
I appreciate that.
Now,
I appreciate that. Okay. So we went over you explained over the embryonic induced pluripotent and the mesenchymal stem cells. What are some limitations that they have that you have seen when it comes to healing and regeneration?
Okay, if you'll put up that other table, the Holy Grail wish list table, there's a list of items that people have generated that they would like for Holy Grail cells to do. So starting up at the top, and I was comparing TSCs to MSCs to iPSCs. Now, Holy Grail wish list, they want the cells to be telomerase positive so they could unlimited proliferation potential. Well, TSCs will do it, iPSCs will do it, and iPSCs and ESCs are basically the same. MSCs won't. They conform to Hayflick's limit. Now, they want cells to be present throughout the entire lifespan of the individual. TSCs, no problem. MSCs, no, they decrease in cell number. iPSCs, it's really not applicable because you can take, what Yamanaka did and reported in 2009 was he took a differentiated cell type, fibrocyte, and transfected 4 embryonic genes, OCT4, SOX2, CMYC, and KLF4, and tried and de-differentiated that cell into a less differentiated cell type, a pluripotent stem cell. And actually, it exactly mimics embryonic stem cells and has all the pros and cons of embryonic stem cells. But anyway, so that's what I'm comparing this to. So anyway, so with respect to present throughout the lifespan of an individual, you could take a 100-year-old, isolate the cells, transfect in the genes, you'd have an iPSC, and then, do their thing. Okay. They want the cell to be invisible to the immune system. Well, that'll work with TSCs because they don't have any self-recognition molecules. They don't appear until you get to the ectodermal, mesodermal, and endodermal stem cell stage. So pluries and toadies don't have self-recognition molecules. So they're, in essence, invisible to the immune system. And it takes about 30 days inside the individual for the toadies and the pluries to get to the germ layer lineage stages. And at that time, they train the immune system to accept them as self. So we've done cross-species transplants with TSCs and PSCs, and then later did transplants with mesos, endos, and ectos, and those cells were not rejected if we waited 30 days before we transplanted them in. If we transplanted them in initially, they would be rejected because they're not self, or even 15 days or 20 days, they would be rejected. But after 30 days, they were accepted as self, not a problem. So I had some of my surgical residents practicing open heart surgery on pigs and goats so we could do cross organ transplants. So what we do, we'd take the endos and the pluries from, say, the pig, inject them into the goat, take the goat cells, same thing, inject them into the pig, and then cross the hearts. That's what we were trying for. We got close, but I got sick, and then we didn't complete it. But that was the idea of being able to do organ transplant, elective organ transplants without having to use immunosuppressants. So that was the idea of the invisible immune system thing. Okay, the next one was they'll form any somatic cell type in the body. Yes, TSCs will do it. iPSCs will do it. Sorry, mesenchymal stem cells only form fat, cartilage, and bone.
Yep.
They don't want the Holy Grail cells to spontaneously differentiate. TSCs won't. MSCs won't. iPSCs do. For iPSCs, and that spontaneous differentiation actually leads to teratoma formation, which is a problem, big problem. To get around that, they take iPSCs or ESCs, as the case may be, and pre-differentiate them to a single cell type. Once that is done, teratomas don't form. Unfortunately, they've lost the plasticity that they had when they were naive. So that's a downside there. But with respect to pre-differentiation, it's not needed for either TSCs or meso-SCs because they don't spontaneously differentiate. They will not form teratomas. Both TSCs and MSCs are tightly controlled by biological agents. MSCs specifically with progression factors and proliferation and progression, and to some extent anti-differentiation. TSCs are controlled by proliferation factors, inductive factors, and anti-differentiation factors. So I'm skipping between different charts and stuff.
No worries.
Okay. They want the cells to produce exosomes. Yes, all three do. Now, do you know why? Do you know what the exosomes do? Most people have no clue what they do. Actually, the exosomes from each cell type, one maintains itself in a proliferative state and also induces the cell above it, as far as the less differentiated cell, to become it. So it has two functions. So, the way we clone cells, actually, cells are very social individuals. They like to be around a group. If you put them by themselves, without that group effect, they'll die. So what we did was to take the conditioned media from TSCs or PSA. We did all the cell types for the ATPSCs, and we did MSCs too, for that matter, and put it on these cells. So one cell in a well, but because we use conditioned media with all these exosomes, tissue-specific exosomes, they thought they were in a whole group of people. So they were, fat, happy, and sassy, and they proliferated perfectly fine. And that's how we cloned them. Other people that tried cloning either started with 3 cells. That was Pittenger, trying to show that MSCs would form fat, cartilage, and bone from a single cell. But they used 3 cells as their starting material, so you got to kind of wonder about that. And then Catherine Verfey started with 10 cells for their MAPCs and showed that they could form endodermal cell, a mesodermal cell, and an ectodermal cell. And you're thinking, start with 10, you only come out with 3, you kind of wonder about that. But anyway, we used conditioned media, and it worked, and we could, and we did repetitive. It's, the whole process is called repetitive single cell clonogenic analysis. And repetitive meaning we did take it out, grow it up, replate it, take 1 cell, grow it up, take 1 cell, grow it up, take 1 cell, grow it up, and then go from there as far as the analysis. So we did analyses on single cell clones of all the different 8 categories. We did analysis on cloned MSCs. We did analysis on isolated but not cloned ATPSCs, MSCs, so on and so forth, and discovered a lot of different things. But anyway, moving along. They can be propagated to large numbers without mutation. Without mutations. Yeah, correct. Okay. We discovered that the cell cycle has a particular to have a cell traverse through its cell cycle takes a certain number of hours. For TSCs, it's about 12 to 14. If you try to cycle them in less hours than 12 to 14, they will form mutations. So you don't, basically. We cycle them every 16 hours, and we can do that by titrating the amount of PDGF in the cultures. We use, nanogram quantities of PDGF, and we titrate them to 16-hour doubling times. So we can double them ad nauseum every 16 hours, and we can get as many cells as you want. I took a human mesodermal stem cell clone, did this technique, and we propagated them out to 690 population doublings. That's 2 to the 690th power, okay?
Yeah.
At certain intervals, and we did a standard 100 population doublings, 200, 300, 400, 500, 600, 690. We checked them for karyotypic analysis. We checked them for cryopreservation recovery. We checked them for differentiation. We checked them for cell surface markers. We compared them to our Z1 population. What we did, we grow them up, aliquoted at 1 million cells per aliquot, froze them down. We used 7.5% ultra-pure DMSO and slow froze at -70 and stored at -70. And then when we wanted to test them, we took out an aliquot, plated those cells along with our doubling number cells, and tested them side by side. And there was no difference at each one of these doubling numbers. And we did some other doubling numbers, I think 256 and, yada, yada, yada. They're listed in the papers because we were doing karyotyping and other things. And on my dad, we did this in 5 different species, mice, rats, humans, pigs and horses. So we know it works. So anyway, PSCs double every, I think it's 14 to 16 hours. So we were titrated to 18 hours. So we're outside the range. And then the germ layer lineage stem cells were about 24 hours. So we doubled it at, or we titrated it to about 26. If you do it sooner than that, you get larger numbers of, and I mean, you can do this with MSCs too. I mean, but they use a factor that makes them proliferate faster than their cell cycle rate. And then they get mutations. Bad thing to do.
Yeah.
You've got to keep them proliferating outside their cell cycle rate. Because the deal is, at the end of division, or near the end of division, you have DNA checking enzymes coming in and checking the newly strand created of DNA to see if it matches the original. If it doesn't, it clips out the bad part, throws it out, recreates the new one, and everything goes. And it takes about, like I said, in TSCs, 12 to 14 hours to do that. If you don't give them enough time for that to occur, the mutations that form stay. And then they go on and on and on. So anyway, that's just a hint for those that are doing propagation to get a whole bunch of cells. You got to know what your cell cycle number is.
Yeah.
Okay.
Intense.
Absence of cell surface markers. Now, we'll get into graft versus host disease in a bit, but the idea is each cell, at least each somatic cell, contains self-recognition molecules on its surface that delineates self from non-self. The immune system looks at a cell. Is it self? Hey, fine, welcome. I'll shake your hand, you're welcome in. If it's non-self, it destroys it with prejudice, literally. It'll chop them up in small pieces. That's the innate part of the immune system. Present it to the adaptive part of the immune system and say, okay, remember these pieces. If you ever see them again, kill them. And that's the, T cells, B cells, that sort of thing. So anyway, they would like the Holy Grail to be invisible cell, to be invisible to the recipient immune system. Well, TSCs will do that because they don't have the recognition molecules. MSCs have the recognition molecules. They won't. Now, with respect to iPSCs, people are thinking, okay, if I take the cell out from the same individual, granted I transvect it to get it to a differentiated cell time, it still should have the same cell surface markers, right? No, wrong. The transvection process changes the cell surface molecules and makes them look different. Non-self. And a lot of studies now coming out, they're saying, my God, I put iPSC cells in the same individual and they're being destroyed. I don't understand what's going on. They changed the cell surface markers. Their immune system is viewing them as non-self. They are foreign. They can have deleterious effects on the individual. I mean, the immune system was set up basically to protect against bacteria, viruses, mold, fungi, drugs, environment, anything under the sun. Anything that's not me gets destroyed. That's the whole point here. So as long as you're self, everything's okay. Everything's copacetic. Okay. They want the Holy Grail stem cell to be used as a universal stem cell transplant. It can work in anybody. Well, okay. Technically, yes, for the TSCs, but there are caveats to this. It has to be gender specific. Unless you want to be a transgender. Anyway, if you transplant female cells into a male, yes, that'll work, but you will see secondary female sex characteristics in the male. Gynecomastia is a big one. Breast formation. Hair pattern is different between males and females, and there are a few other things. If you transplant a male into female, again, secondary sex characteristics. Basically, they get mustaches and beards, but they also have increased musculature increased bone because males have a different framework than females do. So anyway, you can't back up. You should not cross genders unless you want the individual to be a transgender. Then it works perfectly fine.
Of course.
Okay. The other caveat is that with respect to the TSCs and the PSCs, they have to be either ABO blood group matched, in other words, A plus, B plus, B plus, so on and so on, or O negative. Now, it's not necessarily the blood groups themselves, but it's the differentiation of those cell types later on that the immune system doesn't recognizes them as self rather than non-self. And that's why you need to match the blood groups. Those are the two caveats with respect to ATPSCs TSCs as far as using them as universal donors. Now, they would like a long shelf life for the Holy Grails.
Yeah.
We've been able to take out the TSCs out to 40 days if you store it at 4 degrees centigrade in either saline or plasma, and they are just as active as day 0 totipotent stem cells.
Wow.
Yeah. Cryopreservation-wise, we can store them at least 20 years, and they're still as active as the originals. That's as far as I've gotten as far as shelf life is concerned. However, one of my sci-fi projects, I have sci-fi projects. One of them is can I freeze dry these stem cells? Now, the problem with freeze drying is the water content in the cytoplasm. Because once you freeze and try to dry, the water forms spicules, little needles, and they puncture the membranes and the cell's dead. TSCs are a different type of cell. It is a very heterochromatic nucleus. I mean, I have diploid TSCs from 5 different species that are smaller than their respective sperm heads from the same species, which are haploid. So they have double the chromosome content that their sperm do, yet they're smaller in size. So they bring new meaning to the word heterochromatic nuclei. But anyway, you have heterochromatic nuclei, you have 2 mitochondria, you have a couple of ribosomes, and then it's covered in plasma membrane. So there's virtually no cytoplasm whatsoever in these cells. And we did TEM studies to prove it. So I asked the guru of cryopreservation, yada, 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 do you think these cells would freeze dry? And he said, yes, based on the criteria you presented, they should be basically the only ones that can be freeze dried and reconstituted, viable. So that's my next project to try. And the guru I'm talking about is Kelvin Brockbank. He has published loads of literature on freeze drying, on transitional drying, on, a lot of information. So if you ever need information on cryopreservation or anything thereof, go ask him. Okay. When we do studies like cryopreservation and hopefully the freeze drying, we look for increased viability as high as we can get. For cryopreservation, our TSCs, we can reconstitute 98 to 99% viability after cryopreserving for 20 years. If you read ATCC, which is the Bible for cell culture, they suggest using their cryopreservative, and what they think is great is 50% viability after thawing. And I'm sorry. No, I want the highest I can get.
Of course.
The best we had, like I said, with TSCs, PSCs, and the germline lineage were 98 to 99%. With respect to the MSCs, using our protocols, we could get a 95% recovery.
Good.
That sort of thing. So again, I try for the highest. So I've set the bar lower for TSCs, for freeze drawing, and I said, I want greater than 95%. Maybe not 99, 98, 99, but 95. If I can get that, I'm home free. Now, the idea of the freeze drawing is that the cells can withstand extreme temperature ranges. Liquid nitrogen, 196 degrees centigrade, up to Sahara Desert or Death Valley, 200 degrees centigrade, this sort of thing. So anyway, they could be shipped anywhere in the world. They can be stored basically on a shelf, shelf in the kitchen. They can be used, reconstituted, used. Anybody can get these cells. And that's one of my goals, is to have, be able to make these cells available to anyone that wants to use them. Okay, so we keep going down on the list. Okay, I guess not. Okay,
It's
you
right
don't
here.
have it.
Right here.
Yeah, okay, so move up a little bit. Okay, temperature ranges can be bioprinted. Okay, the other thing is, as a freeze-dried cell, it could be bioprinted into 3D constructs for transplants. Now, one of my sci-fi projects, again, is what I call butcher paper. If you've ever gone to the butcher, they have this big roll of white paper. You have a slab of meat. They tear it off, wrap it up, give it to you. Okay, my idea of butcher paper is the extracellular matrix with embedded freeze-dried TSCs, also freeze-dried. So you have the sheet. Now, okay, they say, okay, somebody comes into the MASH unit, the operating room, and says, okay, we have this victim, they have compound fracture. They have, we have to cut away some muscle. So what do you need? Okay, starting off, I need some bone. Okay, so they rip off a piece of tissue, sprinkle it with BMP, which induces bone formation, wrap it up and connect it to the ends of the bone. And then say, well, okay, I need perichondrium. Okay, well, you take another sheet, sprinkle it with perichondrial morphogenetic protein, wrap it around the bone, stick it in. Okay, I need a blood vessel to connect, I need an interim blood vessel to connect. Okay, well, you take another sheet, sprinkle it with VEGF, vascular endothelial cell growth factor, and acidic fibroblast growth factor, which induces endotheliogenesis, blood vessel formation. Wrap it around a pencil and connect it to the blood vessels. I need a stretch of nerve to make the connection, or I need a stretch to connect nerves. Okay, so nerve growth factor on your little butcher paper and connect it, wrap it in a little circle, connect it, on and on and on. Then you send the patient out, and within a month to 2 months, all these tissues have reformed. That's my idea of how to regenerate limbs, so to speak.
How close have you gotten to that?
It's sci-fi. Well, right now we're down to the point of almost ready to freeze-dry the stem cells. So I'm getting there, not quite. I've got a lot of sci-fi projects I want to work on, but I want to get these cells out to the masses first.
Yeah.
That's the most important to me.
So where are you when it comes to getting those cells out to the masses? Where are you in that? Because that would be your first
we've sent ATPSCs to Japan. We've sent them to France. That's why I know how long they'll survive at 4 degrees centigrade for people to work on.
Okay.
That's for research purposes. If you want to do them for treatments, we have worked out the procedures for isolating them from people. And we'll get into that, I think, in episode 2, as far as how we do it. And I'll tell you most of the specifics, not all of them, but most of them.
Yeah.
At least to get you started and this sort of thing. So I know we've skipped around your thingy a lot, but your outline, but and what other questions would you like?
Yeah. So we did skip around, but they were answering multiple questions from everywhere. So you went deep into what ATPSC is, and I know the video, for those that saw the video, obviously it was a great video, but I would like to hear it from you, right? So like in basic terms, what are ATPSCs? And I know you explained, and then they have three different words, three different parts of it: the adult, telomerase positive, and stem cell. What does each of those mean? And why does each of those three words matter?
Okay. Adult means we can isolate the cells from someone after birth. So, and they call postnatal individuals adults, even in this context, adolescents, pubertal, sexually immature are all considered adults. Yeah, whatever
Okay.
terminology you want to use. We can isolate the cells from newborns, pubertal, sexually mature, mature, geriatric individuals,
Okay.
humans. I mean, we published
Human.
those
Yeah.
studies in the early 90s. Not a problem. So we can isolate them from adult individuals. Telomerase positive, all of them are telomerase positive. The toadies, the pleuries, and the germ layer lineages. Although in culture, which we'll get to in the second episode, they look different. They perform differently. They grow in different conditions, and I'll go over those. That's not a problem. In fact, I've actually published them. I published all the protocols in 2025 and have tried to put them on LinkedIn. And I think, I don't know, they may have been too intense, dense, or whatever, but it just kind of right over the top of the head. So I'll sit down and I'll explain everything that I can, that I've published, that's in public domain, let's put it that way. And then as far as what's the last one you had?
Telomerase positive and then stem cell.
Stem cell. Okay, it is what I call the true stem cell. It is pre-programmed to heal. That's its only function. It's not to support functional cells. It's not to form progenitor cells unless there's tissue damage. Then it will go in and yes, it'll form differentiated cells. It'll form progenitor cells, but only in that case. Otherwise, and that is healing, repair, restoration, regeneration. It's not maintenance. It's not function, this sort of thing. So I call them true stem cells in that respect.
There's a T in there, so yeah. In the video, we mentioned about the Hayflick limit, right? Which is
70 population doublings. Yeah.
Yep. So can you explain that a little bit? What's the limit, and why do most adult stem cells only divide a certain number of times?
Okay. Leonard Hayflick in 1962 reported that he took adult human fibroblasts and put them in culture and could have them double maximum 70 times. And that was for newborns. If he took 30-year-olds or 40-year-olds, they would only double like 50 times. But there was always this 70 population doubling cutoff, and that was it. So that was called Hayflick's limit. That is for cells that are not telomerase positive. So that means progenitor cells and differentiated cells, which are not telomerase positive, have this 70 population doubling biological clock. Now, the problem is that if you have any adverse lifestyle, let me back up. The biological clock should match the chronological clock, meaning if you're 10 years old, your organs should be 10 years old. If you're 20, they should be 20. If you're 30, so on and so forth. The problem is with adverse lifestyle. If you have an adverse lifestyle that can impact a particular organ, that organ proliferates faster. The progenitor cells and the differentiated cells proliferate faster to keep normal function intact. But their biological age increases compared to the chronological age. And the examples I could give would be if you're an alcoholic, that impacts liver. So the liver would have a biological age older than its chronological age. If you smoke or vape with nicotine or use any type of nicotine, well, let's just do the smoking and the vaping, you're impacting lung function. So your lung tissues have a higher biological age than your chronological age. Now, liver will too, but lung will repair itself, but it takes 2 pack years for every pack year that you smoked. So if you're 45 years old and have 30 pack years of smoking and stopped, key is stopped, it'll take you 60 years to regenerate your lung. Liver will regenerate too, but again, you got to have the cells present. And the bad thing is that alcohol or smoking will kill adult telomerase positive stem cells that are dividing. So they're activated, they start dividing. If you still continue your adverse lifestyles, smoking or drinking, they'll kill the stem cells. You'll never have the ability to repair the tissue. So you got increased damage, inflammation, chemokines, process, stimulate more proliferation, continue to drink, continue to smoke, kills the stem cells. So this process keeps repeating and repeating and repeating and repeating and repeating until you exhaust your telomerase positive stem cells. And I know say you can't exhaust them, but if they're proliferating, you can kill them, and that will exhaust them.
Will like high levels of stress and like mental, you're not mentally strong, does that also play a big role into that, or no, or just adverse lifestyles like smoking and drinking?
Well, okay. We have an informed consent guideline that we put out when we do we're under IRB approval. So we have an IRB committee that looks at our protocols and makes sure we're not trying to harm the patients in the process. And we have this informed consent guidelines the person has to look at, and we go through what they should do and what they should not do. And no alcohol, no smoking, no lidocaine. The local anesthetic lidocaine kills telomerase positive stem cells, and no chemotherapeutic agents because they kill. You have to limit caffeine to less than 95 milligrams a day, which is equivalent to 1 cup of coffee a day, up to about 2 weeks before we do a transplant, because caffeine inhibits the differentiation of the cells. Prednisone. It pre-differentiates toadies and pleuries to the mesodermal germ layer lineage. If you want mesodermal tissues, no harm, no foul, not a problem. But if you're trying to treat Parkinson's, Alzheimer's, or say diabetes, that's ectoderm, endoderm, it won't work because you've prevented them because you've already pre-differentiated cells to a different lineage. And we have a whole plethora of things that you should and should not do. So we present these to the individuals that we treat before we ever go in and say, you have to follow these rules. If you don't follow these rules, it isn't going to work. Don't waste your money. Don't waste my time. Some people have agreed to it. Other people haven't, and they still smoke and drink and failure. And I mean, we had 7% failures in our treatments based on people that continued adverse lifestyle choices. Yeah. We had 7% of our failures due to the body saying, oh, I want to use those activated stem cells for something else. In a lot of cases, it was to treat cardiomyopathy of the individual, which we didn't know about at all. Individual didn't tell us. They said, well, I want to be treated for Parkinson's. And, okay, we'll do this. Well, they grabbed the cells that we'd put in the brain, took them to the heart, and repaired the heart instead. Because it didn't treat the original problem, we had to list it as a failure.
And was that, and did you guys, how did that, well, now I have a couple of questions. How did that happen? Like, did you know about their heart problem before or no?
No, no, we did not.
Interesting.
The first Parkinson patient we treated, uh, you know, okay, we treated him for Parkinson. We did an intranasal to the, we'll get into this later, but we did an intranasal to the nose with TSCs and PSCs and germ layer lineage IV. 6 months later, the son called up and said, Dr. Young, I have some great news. And I said, have the Parkinson's symptoms, reversed? And he said, no. And I said, oh, what's the good news? He said, my father had a myocardial infarct 25 years ago, or some number of years ago. And his, I guess it was that his cardiac output was 25%. It's raised to 50% or raised to 35%. I said, okay, this is when we were first starting out and we didn't know this stuff. And then I said, do you want a second treatment? Sure. So we brought him in for second treatments. And
now for the cardiac now, not for dementia, for Parkinson's, you said.
They wanted dementia. So we did dementia again. And again, the body stole those cells and treated the heart instead. So they were up to 50%. And I said, do you want us to treat for the dementia? And they said, no, we're happy with our father the way he is. And I said, okay, whatever, Then we learned that.
Forget everything, but as long as he has a good heart,
Yeah, hey, whatever, So we learned that we got to know the background of these people more than what they're telling us. I mean, we were treating COPDs. I had 51 patients that I treated, or my group treated. I can't treat on PhD. Yeah. But anyway, 51 patients. One of them came from overseas. He was kind of frail looking. And we said, okay, exclusion criteria are you can't have active cancer, you can't have inactive infectious disease, yada, yada, yada. No, I don't. Okay, so we went ahead and treated him. Later, we found out that he had AIDS, that he had active AIDS. Okay, we are processing blood products. Okay, yeah, eyes wide open, yeah. We follow universal precautions. We're protecting the cells. We're protecting the cells from us and us from the cells. So, in essence, no harm, no foul. But still, it would have been nice to know that he had AIDS before we started. And we corrected his COPD, and actually his AIDS disappeared, which was an interesting observation. Yeah. Okay, so, we have these interesting observations we've seen. We just haven't followed up on a lot. We followed up on some, but not on all of them. And AIDS is one of them. Can we use, in this case, I wouldn't use autologous, I'd use allogeneics, and treat people to get rid of their problem. Just to be sure that you don't have a reservoir of the infectious disease in even the stem cells that you would transmit back to the individual. So if you use allogeneics from someone that doesn't have the disease, you're clean. So we'd use TSCs and PSCs only because, again, ectos, mesos, and endos have the cell surface self-recognition molecules. So, I mean, it's just, again, you've got to know your model system, what you work.
That was the 4 things that your mentor, director, what was he? Told you?
Well, he was my PhD or my master's degree chairman. You got to know your model system. And yes, you do. You got to know what you're working with. And so we've updated our IRB application materials to know what is your past medical history? What is, how about your parents? What did they have? Did they have any diseases that could be transmitted genetically? How about your siblings? How about your aunts and uncles? This sort of thing. So we know what their background, we want to know labs. If you look at labs, you can say, okay, if you have increased liver function, that means something's going on, this sort of thing. Or if you have increased white blood cells, you've got inflammation going on. Why do you have that? So we'd like to know exactly what we're walking into.
Of course.
Of course. And Go ahead.
I think that can be translated not just in science, but everything, right? You want to know if let's say if you're going to start dating someone, right? You want to know who her family is, right? Like what's her past, you know? If you're going to remodel a home, let's say, right? You want to know how's the structure, right? You have to know everything before you get into anything. I mean, at least, that's what
Yeah,
I would do.
exactly. Exactly. Well, it's the same for medicine. Even though I'm
Yeah.
a PhD, I am geared towards medicine because I taught at a medical school for almost 30 years. We trained medical students in a problem-based, case-based analysis program. We did not lecture at the school. So we come in with a certain case. I was in the phase B, which is embryology, genetics, and cancer phase, introduction to those types. And I was also in the musculoskeletal phase, which is anything dealing with the musculoskeletal system. So we'd have a case, read it, and then say, okay, what don't you understand?
Yeah.
So let's start. What's the normals? What's the normal anatomy? What's the normal embryology? What's the normal, all the different disciplines? Okay, what's the pathology? How does the pathology interact on these different systems? This sort of thing. And then,
You just reminded me. I had a question. So before you were talking about that lidocaine inhibits ATPSCs, I would say something like along
it
those lines.
kills them. It
It
kills
kills them.
100%
Okay.
kill ratio. Yeah.
Okay. So from in orthopedics, I know that some doctors they inject lidocaine before something. So does that inhibit at a local level ATPSCs? How does that work?
Yes, it does. I had a scientist say, well, I use I use mesenchymal stem cells and lidocaine works perfectly fine. And I'm saying, good for you. But for me, lidocaine kills the cells. So we use bupivacaine as our anesthetic because it has a 0% kill ratio. And the same scientist came back and said, but whatever that is, had a hard time pronouncing things. Kills mesenchymal stem cells. And I said, okay, then don't use it. Easy enough. But yes, lidocaine can inhibit, actually kills, dividing ATPSCs. So don't use it. Use another. And we did, and I published anesthetics, which were 100% kill ratio, which were 0% kill ratio, what were in between. So just, look at the paper and you know what to use. This sort of.
Okay. So I know that I believe in that flow chart that went side by side. I don't remember if we went over it, but the next question that I have is what is the difference between a telomerase positive cell versus a telomerase negative cell?
Well, one has unlimited proliferation potential because of the telomerase enzyme. Telomerase negative doesn't have the enzyme, so it conforms to Hayflick's limit. 70 population doublings, and then the cells croak, basically.
Gotcha.
Also, because of Hayflick's limit, they decrease in number with increasing age of the individual. And it should be increasing biological age of the individual, not per se chronological age. Because you're using up your progenitor cells and your differentiated cells.
Interesting. Yeah, I remember I was talking about how I put a post, something about bone marrow being used for intra-articular injections, right? And there was a physician that was talking about how a 70-year-old doing bone marrow probably, I'm sure there's benefits to bone marrow, right? And other things like that. But I'm not trying to get off topic, but
no, that's fine.
The point that he was trying to say is that, our stem cells do not age as we get older, or the same 70-year-old stem cells, you'll probably have a better outcome with doing a bone marrow in a 30-year-old versus a 60, 70-year-old, right? Well, depending now if they're healthy or not, right? How does that correlate with the what is it called? Hayflick effect.
Okay. You age, decrease numbers, don't get the same response. Now, okay, know your model system. What is in bone marrow? Okay, you have bone marrow stromal cells, you have hematopoietic cells, okay? Number one. You have marrow stromal cells that support hematopoietic stem cells. Okay? You have, because it's in bone, you have osteoblasts. You have chondroblasts. You have adipoblasts, fat cell, fat progenitor cells, as well as fat cells. You have endothelial cells. You have lymphatic cells. You have, our cells are present too. The ATPSCs are there in very low numbers, but they're still there, and we've published it. You have about 18 different cell types in bone marrow. Most of them conform to Hayflick's limit, meaning 70 population doublings, and they croak. But now we're talking, this 70 population doublings means you have one cell, it doubles. So you have then two cells, each with 69 population doublings. They double, so you have four cells, each with 68 population doublings. So it's an exponential. It's not just one cell and I'm going to die type thing. But they do decrease with increasing age of the individual. And that's why a 30-year-old does better than a 66-year-old. Now, if you isolated the ATPSCs out from, say, a 10-year-old and a 92-year-old, they work exactly the same.
Interesting.
But you have to increase them in numbers enough that you get a biological effect.
Interesting.
So we can do that in culture, ex vivo. We can do it inside the individual. We use CNSP. We do the fresh isolates with nutraceuticals, so we can do it artificially, various ways to do it. And yes. I think that's in the second episode. So
yes. So then, yeah. So then I know that ATPSCs are found in almost every connective tissue in our body, right? And then you mentioned that you use a nutraceutical CNSP to increase that or something like that. Can you explain that a little bit?
It's okay. To do the background for that story, I had a friend who had leukemia, and they treated him with various cocktails of chemotherapeutic agents. Cured the leukemia, but trashed his heart. So they went in and did 5-way CABG procedures. CABG stands for coronary arterial bypass grafting. So they did 5 of those, plus 15 drug-eluting stents to get the blood vessels open to get blood to his heart. A week later, he had a massive heart attack. Left him with less than 10% cardiac output. He couldn't walk 10 steps without passing over, keeling over. His physicians put his name, his cardiovascular physicians put his name on the heart transplant registry for a heart transplant. And they basically, in my mind, sent him home to die because he was 60 years old.
Young.
Yeah, not young. If a heart matched him but matched a 30-year-old, it would go to the 30-year-old person. It's this sort of thing. They had a longer lifespan.
I see what you're saying.
Okay, so I said, Henry, please let me do we were in the fresh isolate trials. That we'd already started those trials. Let me have fresh isolates. I said, no, I absolutely refuse. Why? I said, we take out for you, it would be 400 mils of blood just to get the stem cells out of the blood.
ATPSCs.
Yeah, ATPSCs.
Okay.
I'll tell you how we do it in a minute, but that's what, that's 400 mils of oxygen capacity that is trashed. You can't withstand that. It would kill you. So no, I'm not going to do it. He said, well, please, 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 let me, think I'm something. So, put the cap on the head, and I said, okay, what can I do? Well, okay, I've posted numerous times on eat blueberries.
Yes.
I'm not kidding. I am not kidding. This is very important. 1 to 2 cups of, and I say fresh or frozen, I'll tell you in a minute why that, blueberries daily stimulates ATPSCs to proliferate in situ. Okay. It's one of the components that we use in CNSP.
How much blueberries you said daily?
1 to 2 cups, fresh or frozen. It's cumulative daily. It's not all in one shot. And I said fresh or frozen because a friend of mine said, I love blueberry pie. Tell my wife that I need blueberry pie every single day, which is cooked. So I said, no, it's got to be fresh or frozen. Because he was already overweight and he didn't need blueberry pie every day, this sort of thing. So it was fresh or frozen. But anyway, they stimulate ATPSCs to proliferate in situ. So they'll increase in number all over your body, not just in one place. So even though they're rare everywhere, if your whole body is producing these cells, you are your own sterile bioreactor.
Yeah.
Okay. We have a second compound. It's a mobilizing compound that causes reverse diapedesis. White blood cells called diapedes, they move out of the blood vessels into the connective, into the tissues. This compound reverses that process and puts the daughter stem cells into the bloodstream. They're not normally present there. But we turbocharge the process, so we get them into the bloodstream, so it's easy to harvest. We don't have to do a bone marrow tap. Yeah. How many people have actually gone through a bone marrow tap? Because people say, oh, it's easy, it's no problem whatsoever, painless, all this kind of stuff. Bullshit. I was a bone marrow donor for a friend of mine when I was a teenager. I was 17 at the time.
Wow, you're a good friend.
He had brain cancer. Our cells matched. So they took bone marrow out of my hips. I was in the hospital for 2 days. And my God, that thing hurt no matter what kind of painkillers they gave me. He had the easy side. He only got the marrow cells by IV. So he made out like a bandit as far as pain was concerned. But it hurts. I'm sorry. So it's not as easy as they say it is.
Sure.
If you think something, I think every clinician should undergo whatever procedure they're performing on their patients to understand what the patient is going through.
I agree.
I require all my collaborators that are physicians to do just that, because certain things happen. And if they happen, we know it's going to work. If they don't happen, we know it's not going to work. They need to feel what's going on.
I agree. I actually had we do trainings, and one of the doctors that I work with, she said, I stopped offering bone marrow once I did it on my own. Once I did it on my own, I saw how much it hurt. I don't want to do that to my patients.
Yeah, exactly. So, I mean, whatever. Anyway, liposuction, any of these, they need to do it on themselves and say, okay, do I want to do this to my patients?
Yeah, I agree.
So having that in mind, I said, okay, our original work was done with skeletal muscle biopsies. Personally, I would not want somebody to go in, cut my skin open, take out a chunk of muscle and isolate the cells. I'm sorry, it's just one of those things. Yeah. So how can I get the stem cells out without doing trauma to the individual? Well, we just move them into the bloodstream, collect the blood, and do our little magic. And that's what we do. This sort of thing. So 2 agents here. Put them into the blood. We do a blood draw. We take out 2 cc or 2 millimeters. Try it again. 2 milliliters of blood per every pound body weight, up to a 200-pound individual, which is maximum 400 mils of blood, which is the Red Cross guidelines for blood donations. It's 400 mils. So we max out at 400 mils. But if a person is 100-pound body weight, we only isolate 200 mils of blood. We have to go to 2 cc per pound body weight type thing.
Gotcha.
I'll talk about it in episode 2 how we actually do it. We come out. We've segregated the cells into TSCs, PSCs, and germinal lineage stem cells. They're separate. We activate them. And then depending on what disease entity we're going to treat, it's different combinations going different places. And again, that's, I think, in episode 2. So we'll leave it to that. And we've gotten good results. We've treated with fresh islets, either autologous or allogeneic. I mean, we've done both. And with allogeneics, they have to be gender matched, ABO group, or O negative universal donors. we've had 86.4% success, 100% safety. Nobody has ever reported adverse side effects from our treatments. And as far as efficacy, we're at 86.4% with respect to fresh isolates. And again, the 14% was 7% people didn't follow guidelines, and the other 7% body treated something else.
Yeah.
And we treated neurodegenerative, pulmonary, cardiovascular, autoimmune, kidney, and orthopedic in
Did
97 different individuals.
did you finish the story with your friend? I don't know if I had cut you off. Did you finish the story about your friend that you had cardiac and then how you ended up with CNSP and blueberries? I'm not sure if you finished the story.
So we created CNSP. I created CNSP. I took our two components.
What does CNSP stand for again?
Combinatorial nutraceutical supplement pill. Now,
can you buy that over the counter?
No. It's purely through the clinical trials we use. So if you want it, you have to get into the clinical trial. And then if you do, we want all this stuff, and then we'll give it to you for free. Yeah, okay. So anyway, I used the two components from the fresh isolates, and I added other components, one that would increase circulation to all organs in the body, another that would activate the homing receptors for damaged tissues, another one that would unmask the receptors for exosomes. We added another that supported a strong innate immune system. It has to be innate, can't be the adaptive side, meaning macrophages, NK cells, neutrophils, monocytes, that system. And then we also added a factor that prevented tissue overgrowth. Now, I've never seen it in 50 plus years, but we added it anyway, just to make sure that these cells would not form cancer cells. And this particular agent, normal cells can use it, cancer cells can't, and it'll kill them. Okay. So anyway, so we called it CNSP. And another patient of ours said, it's not a supplement pill, it's a super pill. You should call it CNSP super pill. So, whatever you want the S to stand for. Yeah. Anyway, so we gave it to him, and he started ingesting it daily. And we found that you had to ramp up the dosage. You can't start off with your maximum dosage. So you start with 1 pill every day for a week, 2 pills every day for the second week, 3 pills, up to your optimum CNSP ingestion. And the optimum is 1 CNSP pill per 25-pound body weight. Okay. And we tested that in horses, 1,200-pound horses. So we were giving them a lot of CNSP. And it worked. They were superhuman horses. Yeah. Or whatever. Anyway. And the conversion to humans matched. 1 pill per 25-pound body weight. Something's going funky with my computer.
Oh, I can hear you just fine.
Okay, it's back again.
Yeah, I can hear you just fine.
So we started taking it daily. 6 months after he took it, he went in to see his cardiologist. And the cardiologist said, I don't know what in the hell you're doing, but you're ever doing. Keep doing it. Why? We're going to take your name off the transplant list. Your cardiac function's up to 35%. I can't explain it.
And all you had done here was just provide him that CNSP pill.
Exactly. That's it. He didn't
change
blueberry.
Was he ingesting blueberries too or no?
Yes, he was. I mean, that just kind of helps the process. But he could have taken CNSP alone and it did it, this sort of thing.
So after his cardiology told him that, what happened after that?
Okay, he continued taking it. At 12-month hospital visit, his cardiac output was up to 50%, which I might add is normal for his age group. People say, well, ATPSCs, I can go back up to 100%. I said, no, it only goes to your age. Whatever your normal is for age group is what it will be. So if you're 80 years old, your normal or 70 years old, your normal heart function is 50%. So that's what will bring it up to 50%. It's only if you use your own cells. If you use someone else's cells, on the other hand, they can get you higher as far as numbers. Our SLE patient had allogeneics, and they brought him up to 70% function.
Interesting.
So, I mean, it just depends on which cell types you use.
Gotcha. That makes total sense.
Okay, so anyway, and at his and now he's still taking them daily. He's off the transplant list. He is playing 9 holes of golf daily, weather permitting. So he's walking more than 10 steps, and he's
Awesome.
doing fine.
Yeah.
And after 2 years on CNSP, both his daughter and his wife commented that, after the chemotherapy, he was bald. His hair is now coming back in his original color.
Wow.
Before chemotherapy, his hair was white. Chemotherapeutic agents, bald. Now, 2 years later, on CNSP, his hair's coming back his original color, which was auburn.
Yeah.
And they're going,
And wow. And is he insane? And then that CNSP, is it for life or what? Or like cycles?
I would take it for life, and I am. I'm in that particular clinical trial.
Okay.
I started in 20.
No wonder you look great.
Oh, thank you. I cheat. I have CNSP.
I would too if I were you. I
Say
would
what?
too if I were you.
I know the technology. I know it works. Hey, I'm all for it. So I'm on 2 clinical trials right now. One is CNSP, and the other are the ATPSCs. But anyway, and it works. But yes, I would take it for life. And we have had, he is still in the trial. He started in 2020. We still send him cases of CNSP free of charge. It's been now, 6 years?
Amazing. And he's still alive and
he's still alive, doing fine, fat, sassy, yada, yada. Although, CNSP, okay, fresh isolates. There are a lot of things you cannot do. You can't drink, smoke, yada.
that's okay. I think that's a good thing.
CNSP, it's not as stringent as fresh isolates. We had one lady that had hip problems, hip pain. So she was going to have hip replacement. I said, let me try CNSP first. Okay. So she started taking CNSP, but she used to have 2 glasses of white wine daily. It still worked. No pain. Easy ambulation. And then her physical therapist started getting her into positions that I didn't think any normal person should be in with respect to hip, and damaged whatever was left, and then she had to have a hip replacement. But before that, CNSP reduced the pain and increased the ambulation on its own. And she was drinking wine. But the idea is you have enzymes in the liver that detoxify alcohol. So you drink alcohol, and then depending on how much you drink, you have a curve. If you go below where it affects ATPSCs, they're fine. This sort of thing. So that's the idea with drinking alcohol, so you can have some, just, if you want to binge alcohol, or if you want to go to a bachelorette party or a bachelor party and you're drinking a whole bunch of alcohol, don't take CNSP for about 2 or 3 days before, and don't stop until 2 or 3 days after, and let your body detoxify all that alcohol, and then you can start up again, this sort of thing. And we had one guy that, okay, no alcohol, no, he said, okay, he came in, he flew in to Charlotte, where I was doing clinical trials before he moved down to Alabama. And we did a harvest and nothing. There was nothing there. And in the post-harvest interview, I said, well, why no stem cell? He said, well, I kept to no alcohol for 30 days. I said, well, what happened? He said, well, last night we had a he owned a business, and I took all my salespeople out, and we kind of drank. I said, "How much?" "Well, quite a bit." And I said, "You killed all your stem cells." That's why it didn't work. And we've had
Man.
others that have gone to bachelorette parties and done the same thing. They're drunk and then no stem cells.
Oh,
It's
okay.
one of those things.
Yeah. So in the video, you call ATPSCs the body's repair network, right?
Yes,
And you also mentioned that you used a term quiescent. Quiescent? They're quiet,
quiescent,
right? Yeah.
quiescent, or dormant, or hibernating. You can
Quiescent.
use all three.
Quiescent. Okay. And meaning that the cells are dormant, they're quiet, right? So how can you wake them up? And if they're so powerful, why are they awake? I mean, why are they
Why
dormant?
are
Why are
they
they
sleeping?
sleeping?
Okay.
Yeah.
Their function is to heal.
Okay.
Okay. If you don't need anything to be healed, why should they be active? No.
Okay.
Yeah. So they
Fair.
just sleep. It's kind of like firemen, things like that, whatever.
Okay. Mhm.
Or emergency service personnel. They sleep when they can, they eat what they can, they do their work, this sort of thing. ATPSCs are the same way. The body activates them with chemokines. They are factors that are released from damaged tissues that migrate out from the damaged tissue area 360 degrees on the next y-axis. So they're up, down, all over our way. And the best analogy I can use is think of a glassy pond, flat, throw a rock in it and you see it, and then you see waves emanating out from where the rock hit, and they decrease in height. And that's the same way with the chemokines. They decrease in concentration as they move away from the damaged site. So the chemokines, once they reach ATPSCs, so if you have a damaged site, the immediate ATPCs in the area, I'll ask you how to back this up. Okay. You cut the skin. Blood vessels bleed. You have fibrin formed. You have platelets trapped in the fibrin. They release their exosomes. You have localized ATPSCs in that area. TGF-beta and basic FGF, which are two prominent exosomes in platelets, induce those cells to form a transitional scar. Forms a Band-Aid, closes off the wound site from the external environment. You have all this dead, damaged tissue, calls in the immune system, specifically the innate immune system, macrophages, monocytes, NK cells, neutrophils. They come in and basically chop up and get rid of the debris and this sort of thing. In the process, they release chemokines. Now, the chemokines they release that we know of so far are PDGFs. They stimulate proliferation. ADF, which inhibits differentiation. And there's also a mobilization agent, which we have not identified as yet.
Okay.
They're moving outward. Every ATPCSC that they see, the PDGFs activate proliferation. So they'll start proliferating. So as you go out and out and out, these cells are proliferating. ADF prevents them from differentiating until they get to the wound site. Mobilization agent, it calls the nearby ATPSCs to migrate through the tissues, through the connective tissues to get to the wound site. The more distant ATPSCs reverse diapedese into the blood vessels, track. They go through the heart, they go through the lungs, and then get to whatever tissues they need to go to, if it's not heart or lungs. So they get there, but they get there at a later time point. Then you have this cascade of events going on, and we can go into it later, or we can go into it now, whatever. That you have the cells there. Progenitor cells are activated by other exosomes within the platelets, and they are IGF-1, IGF-2, and insulin. And they push or accelerate the differentiation of those cells into differentiated cells. Then you have other ATPSCs coming in, and other exosomes within the platelets induce them to form tissues, plus the surrounding tissues release exosomes that say, hey, I want more skeletal muscle formed.
Yeah.
Connect it to me. So that's what they do. Hey, I want blood vessels. Hey, I want nervous tissue. Hey, I want, whatever. They're released from the surrounding tissues, and you have normal regeneration. The exosomes provide the blueprint and the stimulus for normal regeneration. The stem cells provide the workers. You add building blocks, which are, vitamins, minerals, proteoglycans, collagens, yada, yada. Ingest them. Body will break them down into their individual units, transport them to the site, and then the body will rebuild what it wants with those particular components. And you get regeneration.
So yeah, so I mean, it makes sense, right? They're if there's no regeneration, then they're not gonna get called.
Okay. Now, what the physician can do is really screw up this process.
Yeah.
Okay.
How?
Well, transitional scar forms. Okay. Now, oh my God, you got debris under here. I got to scrape that out. So they remove the transitional scar. They scrape out the debris, creating more debris in the process, I might add. You can't scrape out without creating, which calls more innate immune system cells in, which they start debriding the, and you got another transitional scar formed, and then it repeats itself, and it repeats itself, and it repeats itself, and it repeats itself. So you don't get regeneration. Or other times, like I had my finger caught in a car door and somebody slammed it and it completely macerated everything from middle joint down. And the physician said, oh, no, we have to amputate. No, I'm not going to amputate. What do you want? I just want to leave it just the way it is. Now let's see what happens. No, we got to amputate.
Nothing was broken?
Everything was smashed. I mean, you had like powder for bone, it's sort of thing.
Okay.
It was just kind of limp. So I wrapped it up, kept it moist, and just didn't do anything. And the physician, no, we got to amputate. I'm like, no, we're not going to amputate. Why? Well, I did these studies with adult salamanders and they regenerate. So I'm going to test this, see if it works. Lo and behold, I have a finger, intact finger. I don't want to show it because it's usually bird finger, but it's this one right here. That regenerated from here outward. I've got a knuckle here, or I've got a joint right there. The only problem is the nerves normally run along the outside of the finger. Mine at the mid joint come in and run on the inside, or the middle part of the finger rather than the outside. So that's the only difference. But otherwise, I can use it, no problem.
And the doctor really wanted to amputate it?
Yeah, because it was smashed. Now you think of people that have damaged legs. What does a physician do? Well, it saws it off, creates a flap, closes it over, stitches it up. Well, if you got a flap there, the tissue can't regenerate. Physical barrier.
Yeah. So what would be the solution to that so it can regenerate?
Butcher paper, bioactive factors, stem cells.
Amniotic membranes, amniotic tissue.
Well, you can. I mean, our cells are present there. So, they're present in every connective tissue of the body. They're present in the placenta. They're present in the umbilical cord. They're present everywhere.
Okay. So the next question I would ask is like, how would you want people to think about the hierarchy of stem cells? Like,
okay.
Pull out the flow chart.
Yeah, pull out the chart. Okay, now let me back up for a minute before you pull out the chart. In 1990, like I said, James Thompson, published on embryonic stem cells. So those were the only ones that stem cells that could help people, adults. But there was Firestorm again, Vatican, right wing, yada yada. You're killing
Yep,
babies.
politics, all that.
Kaplan published the mesenchymal stem cells and say, okay, here are adult stem cells, but they have some problems. But they're progenitor cells, but everybody jumped on and say, oh, those are adult stem cells. We now know they exist, whatever.
Yeah.
So now there was comparison between the two. Embryonic stem cells, mesenchymal stem cells. And then Yamanaka did the iPSCs, which mimicked the embryonic stem cells. So, they were plastic as long as they were undifferentiated, but you had to pre-differentiate them before you could transplant them. So they're same problems as embryonic stem cells, but you weren't killing babies. That's the whole point here. So back and forth, this sort of thing. So now for the hierarchy, this is the chart that you got is a summation of unidirectional differentiation. And I say unidirectional because it only goes one way. In this case, left to right. In the other one, it was top down. And we did staining, we did sizing. There are different sizes as you go across. For the progenitor cells, the mesodermal cells are 15 to 30 microns in size. For the others, they're actually larger than the germ layer lineage stem cells. So, that was the whole idea. We also looked at trypan blue staining. Now, according to ATCC, trypan blue will distinguish dead cells from live cells. So anything that is trypan blue positive is dead. Anything that's trypan blue positive is alive. Well, what they really don't understand is plasma membranes are very leaky. And the cells, PSCs and higher, have a pump that pumps out the dye. Well, you have to be alive to pump out the dye, so they're telomerase negative. Trypan blue negative. Totipotent stem cells, halo-like stem cell, or HLSCs and CLSCs. Okay, totipotent stem cells don't have the pump. They are always trypan blue positive, but they're still alive. And the little joke I used to tell about during my seminars is there's only two entities that I know that trash increases exponentially, quote unquote trash. Totipotent stem cells in my teenage daughter's bedroom. And I get a little chuckle out of that. I said, but they're live. They increase in numbers, and they're trypan blue positive. The HLSCs and CLSCs are transitional cells. HLSCs are closer to TSCs with respect to staining characteristics and cell surface markers, but they're pluripotent. They're not totipotent.
Gotcha.
CLSCs are closer to PSCs with trypan blue staining and cell surface markers, but they're not. They are pluripotent. And then GLSCs are a transition state between ecto-meso-endo SCs and the PSCs. So they have attributes that are similar as far as cell culturing is concerned, but they're still pluripotent. And they're also trypan blue negative and this sort of thing. And then the ectos, mesos, and endos are larger in size, trypan blue negative, and have particular cell surface markers. And they also express telomerase. Then there's a division point. There's loss of telomerase enzyme. So the cells can form the Hayflick's limit, 70 population doublings before they croak, and they form the progenitor cells, ectodermal, mesodermal, and endo. And then those go on and form the differentiated cell types. So it's
This is a great flow chart. This is a great flow chart. This is a great flow chart. The way you explained it now, like I'm understanding it better now.
They basically recapitulate embryonic development. Now, I also have another flow chart, and I'm not sure you have it or not, is the lineage map, which shows the differentiation from the zygote down to the differentiated cell types. So if you don't have it, I'll post it for you. But anybody that,
No, I said, yeah, okay.
Okay. And I posted it on LinkedIn. And I'm sure it went over the head of a lot of people. But the whole point was you have to know what your cells will form. You can't use a mesodermal stem cell and expect to form neural tissue
Yeah.
or pancreatic islets. It doesn't work. It's a different lineage. And that's what that says, and that's what this says as well.
I understand that. Okay.
So people have said,
Why do you?
I've used this. Go ahead.
No. You go ahead. I was going to ask a question, but go ahead. Just.
There are reports in the literature of saying, well, I isolated mesenchymal stem cells and they'll form neural tissue. And I'm saying, your cultures are contaminated. What do you mean? I said, your cultures are contaminated. But I isolated the cells the way they told, I said, I'm sorry. MSCs co-localize with ATPSCs anytime there is fat involvement, hyaline cartilage involvement, or intramembranous bone involvement. So in those same areas, MSCs and TSC and ATPSCs co-localize. So say if you isolate from adipose tissue or umbilical cord or bone marrow, you're going to get both populations. Now, granted, my cells are significantly less in number than yours. I mean, they're like less than TSCs are like 0.01% compared to progenitor cells that are like 49% of the cells in the body. But they're there. However, your cells double at a very slow rate. Mine double at a very quick rate. So, if you double your cells and it takes 48 hours to double, my cells have already gone through 4 population doublings or more.
See.
So in time, we'll get more of my cells in your population. And I don't believe you. I don't believe you. I don't believe you. I said, believe whatever you want. Okay. We tested it. We took our clones, our TSCs, PSCs, and ecto, meso, and endo, and we genomically labeled them. So we could track them both in culture and in vivo for preclinical animal studies. I took meso-SCs and cloned them. And what we did was to take 1,000 MSCs and put in 5, either one of the 5 cell types, and let them grow up. And then we tested them with our battery, with our library of biological factors. Any time that we saw anything that was not white fat, hyaline cartilage, or intramembranous bone was double labeled with the genomic label. Okay. Then we looked at mixed population. We forced mixed population.
Yeah.
The next thing we did was to take isolates based on what was presented in the literature. And we looked at, and we tested it with our library. And any time that there was ectoderm or endoderm lineage cells, or even some of the mesoderm lineage cells, they were double labeled with the particular cell surface marker for animal, CCAM1 for TSCs, SSCA4 for PSCs, and Thi1 for meso, or for the germinal lineage stem cells. Those are the animal markers, cell surface markers. They co-labeled with the differentiation marker for the particular cell type. Mesodermal stem cells didn't. And we had markers for those. I'm sorry, guys.
So you're saying that mesenchymal stem cells, they fall in the mesodermal?
They are mesodermal stem cells. Yeah, they're the tripotent mesodermal stem cell.
The tripotent because they're into.
Now there are multiple tripotents, but I use mesenchymal stem cells because that was what my cells were being compared to.
Yeah.
But you have,
And I think there's also a lot of people have heard of mesenchymal stem cells too, MSCs.
Yeah, most definitely. It all depends on where the information is published. In the United States, most people read Science, Cell, Nature, PNAS. And with the thought that, well, if it's important, it's going to form, and also JAMA, Journal of American Medical Association. if it's really important, it'll be found in those journals. I don't care about anything else. I don't have the time. Well, unfortunately, all 5 of those journals are political. If you know somebody, like you know the editor, there's a good chance you'll get your paper in. If it matches the narrative of the politics, you'll get your paper in. On and on and on. I found that, well, my papers were rejected from Cell, Nature, Science, and PNAS because adult stem cells don't exist. So I had to publish in 2nd, 3rd, and 4th tier journals. The 2nd tier journals were like Anatomical Record and Cell Biochem Biophys. 3rd tier journals were like Journal of Tissue Culture Methods. Now that's an interesting journal. You submit a manuscript. They send it to a reviewer. The reviewer follows your methods and materials exactly. If they can't repeat your results 100%, the manuscript's rejected, or they suggest revisions. We submitted the ELICA paper and the isolation paper to them back in 1992. Neither paper were rejected. They were accepted without any revisions whatsoever. That's the strength of our techniques. The 4th tier journal are, a lot of people have called them predatory journals because for a price, they will publish your research, but they also have reviewers, but they're not as stringent as the reviewers say that you would find in Science, Cell, Nature. And those people are more looking at who do you know, how's your lab involved, how many NIH grants you have, how many people you're supporting. Does this make sense?
Politics.
Does it? Yeah, it's politics. I'm sorry. It is.
I agree with you. And then I know you last I think a couple of days earlier this week, you mentioned that a lot of also researchers and doctors are biased depending on the product and whatever they're studying. And
of course. I mean, I'm biased to my cells, but I mean, I'll at least look at the other cells and test them. And I mean, what we do in our experiments, we run the ELICA, we do a 96-well plate ELICA, so we can run 12 experiments with a sample size of N or 8 experiments with a sample size of 12. So, I mean, we got a lot of repetition in just a single plate. And what I do is, at the time when I was at the medical school, I would do an experiment. My researchers would do the experiment, and my research assistant would do the experiment. And we would use the same reagents, but made up by different people. Okay. If the results matched within 5% error, 5%, standard deviation, not error, standard deviation, we published, and this sort of thing. A lot of people, one experiment, single 35 millimeter dish, published. No, I mean, I held my research to higher standards and this sort of thing.
That's good.
And again, eventually, if I want to use it on my family members or myself for whatever reason, I want to make sure I'm not hurting them. And that was the impetus for a lot of my studies.
Of course. Well, why do you think a lot of people haven't heard of ATPSCs? Do you think it's because, you mentioned politics, you gave the story about the journals, right? That it's kind of, why, if the science is there.
It doesn't fit the narrative.
Simple as that.
I mean, my collaborator, when I initially started out at Mercer, Paul Lucas is his name. He's the editor of PLOS One. We went to Stanford to see a particular individual that deals with hematopoietic stem cells, and I won't mention his name. Yeah. But anyway, we gave our dog and pony presentation. He said, "Excellent work." I said, "Oh, well, what do you think?" He said, "Your stem cells don't exist." I said, whoa, come on. Well, what did you say? I said, "We just spread," and he said, "They don't exist." I said, "I don't understand." He said, "I have a company based on embryonic stem cells. If your stem cells existed, nobody would want embryonic stem cells. So your stem cells don't exist."
Mm-hmm.
Now, okay, that was lesson number 1. Lesson number 2 was Maurice Radachak in University of Kentucky had VSELs. And we had published on TSCs, which were 0.1 to 2 microns, and PSCs, which are 68 microns. So he said his VSELs were 3 to 5 microns. And they were pluripotent, and they could be formed from, isolated from bone marrow. There were kind of population of mesenchymal stem cells, which is, didn't make any sense. And they could do all this. So this particular individual had one of his postdocs look at Radicek's papers and repeat and said, I can't find them. They don't exist. I looked at the guy's paper, the postdoc, what he published. I couldn't find anything based on what his protocols were. But because it came from a lab that was well known in stem cells, and they couldn't find them, my God, those cells, he's crazy. Those stem cells don't exist. He was trying to commercialize VSELs at the time. They were directing competitors to the companies that this individual had.
Yeah.
So I may be for that, but I'm sorry. I tell it like it is. I've been through a lot of problems, and it just is the way it is.
How do ATPSCs compare to Muse cells, right? Because I know Muse cells, they're also in a very small numbers. And where
Exactly.
Yeah, what are...
Well, Muse cells are a variation of mesenchymal stem cells. They are stress. They I don't know how to put it. They can live through stress. So can our cells. I asked Dr. Khan if he was working with purified clonal populations, and he said, no, our populations are mixed Muse cells. And so, okay. So then I contacted the company that actually sells. He said, we buy them third party. And I said, so I contacted the company that sells Muse cell, actually sells Muse cells that were isolated by the technique of Mari Tozawa. And they said, we don't sell anything to Dr. Khan. I went back to Dr. I said, well, what are Muse? He said, well, they're a mixed population of cells that are stressing to, you're like, okay. So I don't know. They're mixed. If they're mixed, there's, and they're not Tozawa cells, even though they call them Muse cells. And I know mixed populations have our cells in them, and our cells will stand up to strange environmental conditions like, 4 degrees centigrade for a long time. TSCs and PSCs prefer anaerobic conditions. They love 5% oxygen. So when we did, we grew up a lady's cells for our ALS patient, and we did it in a biospherics unit, and we lowered the oxygen tension to 5%, had 5% CO2 and 90% nitrogen. They just went bananas as far as growing. You have to do them in a closed system like biospherics to maintain sterility, but you can't add antibiotic, antimycotic, fungizone to things like that because you're going to put them in people. So they have to be sterile, but can't have the extra agents. So we did it in so a little hint for biospherics unit. And we grew them there. We treated her. She was originally diagnosed in 2013, ALS Laos, LAUS, laboratory. I can't remember what the last one is. But anyway, ALS, amyotrophic lateral sclerosis, that's a death sentence. 3 years after diagnosis, person dies. It's one of those things. She got diagnosed in 2013. We started the stem cells in 2015. We started first with our own cells, fresh isolates, and then it got to the point that we needed more cells. So we went to donors. And I said, I would like 60 or 70-year-olds for the donors. Why? Because they follow, things to do and don't. No, we're going to have 20-year-olds because literature says 20-year-olds have whatever. So they chose 3 females. The first one went to a bachelorette party the day before
hungover,
the harvest, and killed her cells. drank alcohol. The second one swore that she didn't drink alcohol, but she brought a can of regular Coke into our post-harvest meeting. And I said, Coke, how many of those do you drink a day? And she said, oh, anywhere from 12 to 14. 12 to 14 a day?
That's a lot.
I know. Caffeine prevents differentiation. It didn't work. We had the cells, but they just didn't do squat. Third one, didn't drink alcohol, didn't take in just caffeine, but it was an avid runner. And she said, since I've been on this CNSP, I've been able to run 20 kilometers a day. And I'm going, what? 20 kilometers a day? I said, well, how long? Well, I only ran 10 kilometers this morning before I came in for the transplant, for the harvest. 20, she used it to abolish stem cells, healing herself. Instead of building them up for us to harvest.
Yeah. Oh.
So
and that's why you wanted 67-year-olds.
Yeah. They follow directions, mostly to the letter.
Yeah.
Unless they have adverse lifestyle change. Oh, whatever.
That's true.
Okay. So the next step we did was cytophoresis. In cytophoresis, you basically can clean the blood. It was originally set up as plasmapheresis to remove immunoglobulins from the blood. So you could do the entire blood volume and clean out all the immunoglobulins. We set it up to isolate the ATPSCs. So we ran our entire blood volume through the cytophoresis machine and got a whole bunch of stem cells. And then we treated her, and it was like 2 steps forward, 1 step back. But we were making advances. And then there weren't enough stem cells in her blood. So then we went to ex vivo. Now, I wanted to grow universals because then I could treat her and everybody else. No, we can't use allogeneics. They won't have the same cell. Well, TSCs don't have. No. It had to be her cells. Okay. So we were growing TSCs in flasks for this lady, and we were generating billions of cells per month for her treatment. She is still alive 13 years later, still with the ALS diagnosis. She's still alive. 13 years after we started stem cell treatment.
Interesting.
Yeah. So a lot of, in diseases that are trying to kill you on a daily basis, you need more and more stem cells for treatment. The more stem cells you give, the better results. Now, I usually work with terminal patients, chronically ill patients with no chronically.
When you said that you usually work with terminal ill patients, and then during clinical trials, so that's why you are able to help these patients.
Exactly.
Do you use the compassionate
yes, we do.
that, and then also the right to try? Are you familiar with that?
Yes, we use both of those.
Awesome.
To allow us to treat our terminal patients are our ALS patient and our SLE patient.
That's awesome.
Those are our terminal patients. The SLE patient is out. We started treating in 2011, out 15 years and still going strong.
I feel like a lot of doctors and a lot of people in the medical community do not know that about the compassionate use and the right to try.
You have to be under IRB supervision to be able to use those. If you just do it out on your own, you can't use that as an excuse.
Really?
So we offer the clinical trials. Now, I offer my services for free. I have done all the isolations for free, but the clinician charges a certain amount of money for overhead, for nurses, for supplies, yada, yada, yada. That's out of my control. I can't do anything about, this sort of thing. But as far as the foundation I work for, which is Dragonfly Foundation for Research and Development, not Dragonfly Foundation in Cincinnati for kids, different foundations, we give the treatments for free. Our side of the treatments for free. Thinking that, okay, the patients have endured a lot of problems. So why force money on them as well? Because I know some clinics offer treatments for $50,000, $100,000.
Okay,
They
then
use
I don't know anyone
minimally.
who can afford
Hmm?
that. Who's gonna
What?
be able to who's going to be able to afford that?
Celebrities.
Well, I mean, other than them, right?
Real.
And other than the 5% or maybe less,
I know, I know, I know.
you know?
So I've kept my collaborators to not charge more than $5,000 per treatment. I think he's up now to $6,000 in Charlotte.
Mm.
Um,
That's not bad though.
no, it's not. And as long as you follow things and as long as your body wants to not, steals them for something else, there's a good chance you're going to get a better quality of life. And that's all I promise. You have to follow these procedures. We have to know what comorbidities you have so we can address them first. And then we can treat whatever problem you want. And if you follow all the rules, I hope, can't guarantee, but I hope will give you a better quality of life.
I agree. Yeah. One thing is that, with all these, you mentioned allogeneic, that one patient didn't want allogeneic because of the graft versus host disease that I know we
Yeah,
haven't gotten
yeah.
into. And so that it's with safety, right? How do ATPSCs, what are their safety? Is there any safety concerns in that with them?
Yes. Well, okay, let's break out the populations. You have TSCs, PSCs, and the germinal lineage. TSCs and PSCs don't have any self-recognition molecules until they turn into the GLSCs.
That's right.
GLSCs have MHC class 1 self-recognition molecule. So for allogeneics, we do not use GLSCs. We only use TSCs and PSCs. For fresh isolates, we use all three because they come from the same person. Now, the problem with graft versus host disease, you have an immune system. It's made up of two components. You have an innate immune system and an adaptive or humoral immune system. Innate is monocytes, macrophages, NK cells, and neutrophils. Adaptive immune system is T cells and B cells. The first portion of the immune system to see anything that's inserted into the body is the innate immune system. And they check for self versus non-self. If it's non-self and they are competent, they will destroy it.
Yeah.
And I might add, with prejudice. They chop it up into little pieces, present it to the adaptive immune system so they can make antibodies, and for later, if it comes back again, they can kill it. This sort of thing. That is if the person has a competent immune system. Now, that's graft versus host disease. The problem is if the person is immunocompromised. So they could be a SCID patient. They could be an HIV patient. They could be a bone marrow suppression patient. They could be, immunosuppressants that dampen both the innate and the adaptive. They're not killed. The cells that are, or the entities that are inserted are not killed, and they can run rampant and literally kill the patient. Sepsis, and you can die within 12 to 24 hours with sepsis. So you don't want to use anything that does not have self-recognition molecules. You don't want to use anything that does have self-recognition molecules. GLSCs have self-recognition molecules. Everything downstream from GLSCs have self-recognition molecules. I know they say, well, MSCs, they can survive the immune system. Have you tested them for MHC class 1? Well, no. Test them. Because we did, and they did have MHC class 1 receptors, cell surface molecules. So they would be destroyed by the immune system if the immune system was competent. But like I said, anything at GLSCs and below somatic cells have self-recognition molecules. Now, hematopoietic cells don't have MHC class 1s, they have HLA-DRs, which are still self-recognition, but it's just specific for the hematopoietic lineage. So when you do a full organ transplant coming out of somebody, they match as many HLA and MHC class 1 markers as they can to make sure that the transplant will hold. Okay?
Okay.
1 or 2 isn't going to work. It's going to be rejected. And there's, a friend of mine worked in an oncology lab doing testing, and he said, they're up to 15 to 20 markers now that have to be matched before they do a transplant. And I'm sure it's even higher than that to get a better response. You have to know your model system. Yeah, keep drumming it into people.
That is important. Okay. So, looking back 50 years in regenerative medicine, where do you see the field now going, specifically with ATPSCs?
Well, I'd like to see more people using them. I mean,
Okay.
in my, we've worked out all the we turned art into science. We originally started, how we do the isolation? Well, they don't fit the ATCC protocols. We had to develop our own protocols. How you do this? Well, we figured it out. How you do this? We figured it out. How it's an assay? We figured that out. Yada, yada, yada. I have them all down. I have the SOPs. That's not a problem.
Okay.
I'd like to
So
see
how
more
would you
people.
get them out? How would you get that out? How would you get it? How would you said you want more people to use it, right? So like
what I'm doing right now, podcasts.
Okay.
We are publishing. I was sick for a while. I'm back. I published a lot of our preclinical work and early clinical work in 2017. I did the clinical trials in 2020. I did the SOPs for how you do them from solid tissues in 2025. I'm publishing now the rest of the studies, like the, contamination studies. And one of my papers says, give credit where credit's due. A lot of these populations that are mixed populations actually have R cells in them, and that's what's actually causing what you guys are calling MSCs. There's no such thing as transdifferentiation, things along those lines, which I might add a second tier journal rejected because no, doesn't make any sense. Those populations are pure, and it must have been a biochemist that reviewed it. I don't know. They don't know the histology. You got to look at your tissues, got to understand what's there, and go from there.
The details to everything.
And the details, yeah. I mean, this thing hangs on details, but we know the details.
Yeah.
That's the whole point. Anybody that wants to collaborate, I'm open for collaboration. I have the patent. It's already been published. It's in public domain. Have at it. Try to isolate the cells. I mean, I had one person call me up on the phone. Well, actually emailed me and said, I looked at the patent. I tried to isolate your cells and I can't find anything. I said, okay, easy enough. First question, how old was the individual that you isolated? Or I said, what were the age of the cells? He said, I don't understand. I said, how old were the individuals that you were isolating the cells from? He said, well, they were healthy 20-year-olds. And I go, oh. I said, okay, next question. Were they healthy? Well, of course they were healthy. I said, that's the worst population to try to isolate ATPSCs from. Why?
Really?
I said, they're healthy. These cells are healed. To heal things. If they're healthy, they're not going to be in the vasculature. I said, the only time you're going to find ATPSCs in the vasculature is if the person is chronically ill, like somebody with SLE, someone with ALS, whatever. If they have an acute traumatic injury, go harvest 24 hours after the injury. If they are a power lifter and have just reached their personal best record, harvest 24 hours after their personal
because
best
the body
record.
is repairing itself, right?
Exactly. And the reason I know power lifters is my daughter's a power lifter. And we did harvest her cells.
Or going back to the story that you mentioned, the lady that ran 10 kilometers or something like that.
Exactly.
Like
exactly.
That.
They were treating their own cells.
Yeah.
So, I mean, they're there, but I mean, it has to be certain instances. Now, we've been able to turbocharge this process by CNSP and then harvest with the fresh blueberries and then harvest with the fresh isolates. But I mean, the body will do it on its own.
Yeah.
But you gotta choose the right model to use. And if you read the literature says young individuals have the highest content of adult stem cells, and they have to be healthy. And what you're isolating are progenitor cells.
It's like right now I've been talking to a lot with Dr. Carla Mazzeo, and obviously she's in the exosome. So most of the things that, because I get a lot, because I'm very vocal when it comes to talking out about allogeneic, and I'm learning myself, and I ask a lot of questions. And one of the things that she's has kind of ingrained in to ask, it told me, is that whenever someone like a doctor or someone asks you like, hey, what type of stem cells to use? Like the first question is, what are you treating, or what are you going to help? Like what is it that you're going to be doing before you even talk about what stem cell to use, right? So the flow chart that you sent, that you shared with me, with us, that's huge, because now even though there's a lot of things that I probably don't understand, but I can go in and kind of make it dummy proof a little bit for me, and make
There
it
you go.
understandable in that. So I appreciate that and your time today. It has been very great, to be very honest with you.
Well, I may stop you for a minute.
Of course.
Since being on LinkedIn, I've had a lot of vendors contact me and say, well, we know you belong to Dragonfly Foundation, yada, yada, yada. Would you be interested in such and such? And this one guy contacted me about exosomes. I said, okay, I'll bite.
Yeah, I mean, it's always good to like listen to what they say, right? Like,
They said, we're selling exosomes. I said, okay, which ones are you selling? They said, we're selling exosomes. I said, no. Which ones are you selling? And I said, what do you mean? I said, well, every cell in the body secretes exosomes. They are specific for particular cells. Which exosomes are you selling? He said, we're selling exosomes. I was, okay. Then you have PRP, people using PRP.
Yeah.
And I said, and PRP is a bag of exosomes. I said, okay.
What do you mean by that? What do you mean by that? PRP is a bag of exosomes. What do you mean by that?
Yeah. All the components of PRP are exosomes. They're little vesicle-bound substances, inductive factors, progression factors, anti-differentiation factors, progression, inductive factors. And I wanted to know, and nobody could tell me what. So we went in, we lysed the platelets, ran the lysate on SDS page gels, came out with 46 bands on Kamasi blue staining. And I'm sure we'd find more with silver staining, but I didn't want to go that route. 46 bands. Okay. Then we probed bands with antibodies to different compounds. TGF-beta, basic FGF, IGF-1, IGF-2, insulin, PDGFs, AA, BB, AB, inductive factors. Then we took recombinant proteins, morphogenetic proteins, cell-specific exosomes in the library and tested against those particular agents. So we used TGF-beta on our ATPSCs, PSCs, and ecto, meso, endo. What did they do? And, we had that table before. This is what they do. They form scar tissue. Basic FGF, what does it do? Induces scar tissue. BMP2, what does it do? Induces endochondral bone formation. And on and on and on down the list for those different bands. That's what platelets do. I know what's in them. Most people that use platelets have no clue. They just use PRP or PRP concentrated or, whatever.
Yeah, there's a big debate right now. Well, not a big debate, but there's just about platelet dose and then the leukocyte rich versus leukocyte poor. Which one is better? It's, it's just, I feel like it's way more nuanced than what that is, the way they make it seem.
It is. It's very nuanced.
And then getting into, the details and all of that. So I know we hopped around with the list, but we covered literally everything that we had on there.
Maybe sure.
Yeah, yeah. We covered, like, for example, you covered it in your stories that you gave us.
Okay, well, one of the ones was like, where are you know
Yeah, you can
MSCs
bring it up. I
located?
mean,
Now I've got them on my little cheat sheet here.
okay.
Bone marrow, adipose tissue, wisdom teeth. They're now saying, oh, MSCs are present in wisdom. Well, our cells are there too.
Okay.
Periosteum, sternum, umbilical cord blood, basically any tissue organ with a connective tissue compartment associated. I did say that, and things
Yes.
like that. Political wise, uh, okay, we have the comparison between ESCs and MSCs.
Yes.
On one side, ESCs are very plastic, forming all somatic cells of the body, ectoderm, mesoderm, and endoderm. MSCs will only form 3 cell types in the mesodermal lineage, fat, cartilage, and
Yes.
bone.
Correct.
ESCs contain the telomerase enzyme, MSCs don't. Now, but on the flip side, ESCs spontaneously differentiate to form teratomas. MSCs are quiescent, just like our cells.
Yes.
They do not form teratomas. ESCs have to be pre-differentiated to be able to transplant. MSCs don't. You can go in as a tripotent. Good luck with, if it's not a, thingy, but not what you're hoping for, but it happens. And to bring up another point, which is very important for clinicians, they have to know the difference. Okay, there is an acronym called MSC. There are actually 3 different cell types that have that acronym. You have a mesenchymal stem cell that is a tripotent cell that will form fat, cartilage, and bone. You have a medicinal secreting cell, also called MSC, that presumably modulates the immune system during regeneration, although from my side, that's questionable. And you have marrow-stromal cells, MSCs, that support hematopoiesis. So the question is, which MSC are you isolating? Now, I went to an FDA conference a few years ago where they were comparing different things, and then patients got up and talked about clinics they went to, or I should say patients, family members got up and talked about clinics that they or their spouses went to get treatment. In one clinic, they were isolating MSCs and injecting them into both eyes the same day, I might add, and it caused blindness. What they found out from MRIs, in fact,
There was
created
something like
cartilage
that.
nodules
Oof. There was something like
in
that
the
that happened
eyeball.
in Florida. They were doing that. I don't
Yeah.
know if that was in
Now,
Florida.
the point being is that they were isolating the wrong population of MSCs. They isolated the tripotent. They thought they were isolating the medicinal secretory. They didn't understand the nuances between the cells and used the isolation protocol for the tripotents rather than the isolation protocol for the medicinal. Now, they should, even doing that, the clinic should have tested the cell surface markers for the cells. MS tripotent is CD90, CD90, CD105, CD123, CD166, and MHC class 1. Medicinals are CD70, CD73, CD90, CD105, MHC class 1. Different cells. Easily able to discern if you did the cell surface marker profile, which you can do with flow cytometry. That's not a problem. They never tested that before they did the injections. Another clinic in Texas, as the case may be, isolated MSCs from fat, didn't follow the procedures, and didn't get rid of the fat before they injected IV. Formed a fat emboli, and it killed the patient. Yeah. You got to follow the protocol, but you got to know what you're working with.
Yeah.
You can do that with cell surface markers. They're out there. They can tell you exactly which cells you're using. Once you know that, you can know whether you can use the cells for what you want or not use the cells for what you want, or use another population of cells for what you want to do.
Yeah. Well, that's where the translation matters, right? From what you guys see to how the doctors, to patients, how it's commercialized, how it's educated, and all of that.
Exactly. Now,
You know,
one thing is they're growing MSCs and iPSCs ex vivo. So they're increasing cell populations, and then they're complaining that they have mutations. Well, okay, you're increasing numbers at a rate shorter than their cell surface times. I understand that. You should understand that. You can titrate with PDGF to whatever population doubling number or doubling rate you want. That's not a problem. This is how you do it. They also say, well, bioreactors, you have shear forces you have to worry about, yada, yada. And, yeah, they don't understand the cells they're working with. Put them in, one thing I posted, I said, okay, for anybody that grows cells in flasks, have you ever put a stir bar in your flask and see what happened to your cells? Because it's a shear force. And I said, one person came back. Yeah, I said, what happened? Well, it killed a cell. Yeah, right. Okay. Why do you put a stir bar or a stirring mechanism in a bioreactor? You're doing the same thing. Duh, right over the top of the head. Whatever. Another was, we use DMSO, ultra pure DMSO, 100% or 99.5%, 99% ultra pure DMSO when we freeze our cells. And they said, well, no, we use 15% or 70% or, whatever DMSO. 70% is what ATPS or ATCC suggests using. And I said, well, I would be curious what the other 30% is. If you use ultra pure, there's no other point there. And then I said, but you have to be careful because DMSO is very exothermic. And they said, what do you mean? I said, okay, here's a test. You take a beaker of water, you put 50 mils in a beaker and you put a thermometer in there. Easy enough. You take 25 mils of ultra pure DMSO and put it in the beaker with stir bar and tell me what happens. They said, my God, the temperature went up. I said, yeah, it's exothermic. So you have to, disperse the cells in the cryobot. They said, well, when I vortex, everything was dead. Yeah, that's too high shear forces. You have to just back and forth, back and forth, 3 or 4 times. You're fine. Stick in the, and go for it. It's just little nuances like that make or break an experiment. People don't want to do that. They want to mass produce, we do 24 vials at a time to freeze because that's what our container holds, for one thing. And you've got 5 minutes max between the time you add the DMSO till it has to go in the freezer. Otherwise, you've killed the cells. If you kill the cells going in, you're going to have zero recovery coming out as far as viability is concerned.
Yeah.
And then how you do the viability? Well, I just leave it on the counter until it, liquefies. I go, no. Put in a 37-degree water bath or a bee bath, doesn't matter which one. As soon as it turns, pour it into, and it's 14:1, 14 mils media and your mill of cells. Centrifuge it, pour off, and that gets rid of the DMSO, and then you can reconstitute and plate your cells. High viability. Little nuances like that we have learned from
your own mistakes or errors or researching. Yeah.
Exactly. That are in our SOPs. This is what you follow. You follow the recipe. I can guarantee the results. If you don't follow the recipe, I can guarantee the results. They're going to fail. This sort of thing. So that's what I'd like to impart on individuals that would like to use the ATPSCs. We have the protocols. We can tell you exactly what to do.
Yeah.
And this sort of thing.
So then if they want to use it though, they have to be in clinical trials to do it? Are they, how does that work? Like if there's a doctor that wants to try it to, what's that process look like?
They need to be under IRB I would request they be under IRB supervision. That way, if something goes wrong, that protects them and protects me.
Of course.
Protects the foundation, this sort of thing. If they want to do it on their own, I'm not responsible for anything.
Yeah, no. I mean, you would want it because then you're collecting data. You have, support from you, from the organization, from everyone.
With IRB, I can publish the data. Without IRB, you really can't, because it's basically anecdotal stuff.
Yeah.
And I want to publish
Of
because
course.
I want, even though it's in, 4-tier, 3-tier, 2nd-tier journals, I'm still publishing. It's out there. I mean, I started publishing my results in 1977, 2 years after I discovered the cells. I'm still publishing today. 109 publications. I mean, they're out there. Just look for my name. What I did was to post all my research to ResearchGate. It's an open access, freely downloadable source. You can go in, ask for a paper, and download it free of charge. You don't have to pay $1,200 to look at the, research paper, as you would if you had to look at Science or Elsevier or one of those journals and this sort of thing. So it's free. If you don't want to do ResearchGate, send me an email. I'll send you the PDFs. I mean, I have them all on PDFs. That's not a problem. This sort of thing. But I want them to use them the correct way.
Yeah.
Not the incorrect way. Now, because they're so plastic, they can cover up your mistakes. I mean, our SLE patient was in a bad way. They had neuropathy, cardiomyopathy, pulmonary fibrosis, kidney failure, and we brought him in for a 3-way transplant, their own cells and 2 donors. Okay? So we got a whole boatload of cells. Cells went intranasal for the neuropathies. They went Thebesian system for the heart. They went for the lungs, systemic for the kidney, for the autoimmune problems. Unfortunately, it is very difficult to get donors, isolator, and clinician all at the same place at the same time. It's like a nightmare trying to do that. So we had set this up like 6 months ahead of time. Everybody was going to be there. I was going to be there. The patient was going to be there. The clinician was going to be there. And the 2 donors were going to be there. Okay. 1 week before we did the harvest, the donor got bit by a brown recluse spider on the ankle. Formed necrotic tissue. Started out, 2 prong marks. And then within a week, it was about the size of a dime, necrotic tissue. You can see the glistening white tendons underneath. By 2 weeks, it was the size of a quarter. Now, we could have said, no, we're not going to do this. But they were in such a bad way, and it took a long time so we did TSCs through the nose, PSCs and TSCs, or TSCs through the Thebesian, TSC, PSCs for lung, and then mesos for the lung. We did, all the others I talked about. A month later, the wound at the ankle had completely healed. And actually, a zone of about 3 inches diameter of normal, he had hemosiderin deposits all over his legs and ankles and feet. So they're very dark brown. About a 3-inch zone around that spider bite was completely clean. It looked like newborn skin. Nothing worked with respect to the neuropathies. Nothing worked with respect to the cardiomyopathies. Nothing worked with respect to the pulmonary fibrosis. Nothing worked with respect to any of the other organs that we wanted to treat. The body stole the stem cells because they knew they were activated and took them all down and treated the spider bite because that was the most life-threatening condition the patient had. They could have died of sepsis within 24 or 48 hours.
Wow.
The body healed that area instead. And said, forget it. You guys will last. He'll last a little bit longer, and then we'll, later.
So obviously, it prioritized that.
Say what?
It prioritized the
exactly. It prioritizes life-threatening injuries are treated first, no matter where you put the cells. The next is it does treatment in reverse chronological order of appearance, meaning the first things that are damaged are the last things to be repaired. So, you know, if I broke my leg and when I was
Yeah, that
10,
makes total sense. You reverse engineer in a
it's
way.
basing on the chemokines and the concentration gradients and this sort of thing. And the analogy is, you know, you throw a rock in, you see this, you know, in the center, you see, you know, concentrate. Then you throw a rock on one side and you see some others going, you know, so you throw a rock on another side and you see this. Then you throw a cinder block in the center of the pond and it, whoosh, that's the traumatic, you know, that's the life-threatening injury. And then it'll pick up the others as the concentration gradients are. And that's what happened.
Interesting. You know what what I really like is your nutraceutical, the CNSP. That sounds
Yeah.
amazing. That sounds amazing.
We have it in 20 different clinical trials right now. Well,
Wow.
separate entities, but we're
Mm,
not testing the same things as we did with the fresh isolates. We have people with acute scoliosis. We have had two. I mean, they're all done now. But the first one was 35 degrees off midline. And through CNSP and fresh isolates and a chiropractor, they're now 0 degrees off midline. We had another one with 10% degrees off midline that was CNSP and a chiropractor. They're now 0 degrees off midline. We've had people with degenerative disc disease on CNSP and no pain. We have a T12 paraplegic that has only had CNSP. We've treated 2 T12 paraplegics, one with fresh isolates and this other one with just CNSP. And the fresh isolates, she had it in her mind that she wanted to walk. And I tried to explain to her that nerves regenerate at a whopping speed of 1 millimeter per day from the point
is
of
that
the cell
fast or
body.
is that considered fast?
No, that's considered slow. 1 millimeter. So you got to measure from where the cell body is down to the farthest point away. So to walk, you're talking about cells in the brainstem going down to the ventral roots and then going out to, in this case, the tip of the toes.
I see what you're
And I
saying.
tried to tell her it's going to take a while. No, I want to walk. Okay. First treatment we gave, the pain physician used lidocaine, killed everything.
Oh,
Couldn't understand. So that's when we did the study on the anesthetics and found out lidocaine had a 100% kill ratio, the others less. And then bupivacaine was 0%. So second time he used bupivacaine. Fine. 2 months later, she got a second treatment was with the new anesthetic. Third treatment was with the new 2 months later. At that point, we had restored bladder bowel function. So she could poop, she could urinate on command rather than having to wear a bag or anything like that. We thought that was great.
huge.
It was in the time frame of basically 4 months. But she couldn't walk. That's what she wanted, so she dropped out. Now, this sort of thing. Now, I have another one that's T12 that because he lives overseas, I can't do fresh isolates on him. So we sent him CNSP. And 2 years now, and he has regained sensation in the quads, in the hamstrings. He can flex his thigh at the hip joint. So we're that far right now. And that's just on CNSP, but it's taken him 2 years. It's a slower process. I look at fresh isolates as a bolus effect, where CNSP is more of a steady effect. You know, it releases stem cells 24/7, 365 days a year, whatever. And they're activated and then they do their thing. This sort of thing. So it's a different way of looking at things. Our power lifter, my daughter, her original coach screwed her up as far as how you do things. And she damaged her wrists and her knees. So we did fresh isolates, got those corrected. and then she's on CNSP. And it accelerated the time periods between personal best records. So they were shorter, the time periods.
I
And
get it.
last year, or maybe it was the year before, she went to Puerto
You
Rico
should
for,
probably you've heard of the now they have what I think in Vegas or something they have the enhanced Olympics now where they're allowing athletes to use
well, that's good.
like steroids. Have you heard of that?
No, I haven't, but
No, yeah,
that sounds good.
yeah, it's like
Actually,
enhanced.
they should have Olympics for transgenders.
I agree with you
Males that are
100%.
females, have them their own set of Olympics. That's fine, but
I
to
agree
mix
with you.
males with females, I'm sorry.
I
But
agree.
anyway, I think it was 2 years ago, she went to Puerto Rico for the, whatever games there were, and she placed 3rd bronze medal in her weight class for powerlifting. So I was like, "Hey, this is good." So, she's in the trial. Uh, Shingles. It's herpes zoster or chickenpox that once you've had chickenpox, it'll embed itself in your dorsal root ganglion and then express itself anytime you're under undue stress. So they now have a vaccine for, an extra vaccine. Shingrix, I think is what it's called. So,
Always something.
hmm,
There's always
always something.
something. There's always something.
Well, one of the family members signed up to get the vaccine. I said, no, we can't give it to you right away. So they got shingles. Then they got the vaccine and they still have shingles. They still have the pain from shingles. And they were like 10 out of 10 for pain.
Yeah.
Started them with CNSP. They're now 2 out of 10 for pain. So while it didn't completely eliminate the pain, at least it decreased it enough that they can function. So, I mean, different things. I mean, we had a lady that wanted to repair her knees. She was 30 pounds overweight and she couldn't, jog without it hurting. Put her on CNSP. Did it repair the knees? Not sure, but she lost 30 pounds. So now she's out jogging and knees are pain free. So, I mean, the body works in mysterious ways, this sort of thing. Her husband, who was also overweight, lost 15 pounds on CNSP. Um, the original 20 people that were in the trial, we asked them to write down what is different from before you started treating CNSP to after. And all of them said, I have increased, the colors are brighter and sharper, which I say increased color acuity. And we started that trial during COVID lockdown. Okay. And it was like everybody's locked down, they're depressed, they have brain fog, all this kind of stuff. Decreased brain fog, decreased depression,
Huge.
increased cognition, more energy. And I think there are a couple others. Now, they are side effects of CNSP, but they're not adverse side effects. They're just something we hadn't anticipated. I hadn't anticipated. That's okay. So, I mean, as a general, now, we're gearing up to do multi-arm clinical trial testing CNSP by itself, fresh isolates, and TSCs ex vivo, comparing everybody against each other. We're using the patient's past medical history as their identical matched placebo control. We'll do linear regression analysis to say, okay, if we stay on the same technique they're on, what's going to happen? And it's, slope's going to go down. What happens with each one of these? Are they going to go up, go down, stay the same, or what? This sort of thing. Right now, we're using CNSP to get the area of the wound, or actually to correct any comorbidities they have before we actually do fresh isolates for what they're trying to go for. Yeah. So, if they do
You're
heart
prepping
problems,
the soil. It preps the soil.
Yeah, we're prepping the soil, cow manure, mushroom, whatever. Prepping the
Sunlight,
soil.
water, adequate everything, nutrients.
So if they have a myopathy, giving them CNSP for a year should correct that because we've seen that. If they have COPD, it should help with that. It should help with IPF too, for that matter. So any comorbidity that the body would consider it wants to treat more so than what the individual wants treated, we're trying to hit that up with CNSP. And we're comparing baseline values before treatment to post-treatment 6 months, 1 year, 3 years, 5 years, 10 years, and see what happens.
Wow, I'm curious to see how that comes out.
Well, I need volunteers for one thing.
I'll probably sign
I
up.
also need the foundation also needs funding, this sort of thing. So,
Yeah,
plus
sure.
minus there. So that's one. Fresh isolates. So we prep the area. Then, if all the comorbidities are taken care of, we should be able to treat what the patient wants treated, and this sort of.
Gotcha.
Now, our
Yeah, because
trials
it's
are
gonna
open
prioritize.
access.
It's gonna prioritize the worst first.
Exactly. So
Yeah.
CNSP should take care of that. And we'll
Interesting.
know by the lab results. And then, let's see where I think I lost my train of thought here with the fresh isolates. But then we'll treat with fresh isolates. And hopefully the patient gets the treatment they want. Now, the Dragonfly DFRDs clinical trials are open access, open-ended. We go by what the patient wants. So meaning, we say, well, we'd like to treat you 6 times, but hey, if you get the results of 2 times, you can drop out. That's fine. That's not a problem. And we'll just take your results and do it to, whatever time frame you have and report it because it's IRB controlled,
Yes.
IRB supervised.
Yeah.
So they can enter the trial whenever they want. They can leave the trial whenever they want, this sort of thing. So it gives access to more people. And again, CNSP is free of charge as long as you're in the clinical trial. So patients don't have to worry about that. They only have to worry about shipping charges. I used to give it away literally for free, but I'm not financially liquid enough to pay for shipping charges now. So they have to do that.
I mean, but that's the least they can do,
I
in my
know.
opinion.
They can do it at home. They don't
Yeah.
have to go anywhere with the fresh
No.
isolates. You have to go somewhere.
Is there any interest in commercializing that CNSP or no?
There was. We licensed it to a company. They were charging $2,400 a case. Yeah.
What's a case? Is that like pills?
12 120 count bottles.
1,200 bottles. That's not that bad though.
1200. No, 12 bottles, 120 count per bottle, per case, $2,400 per case.
Wow.
Yeah. Pricey.
Yeah. So what? That's only 12 bottles.
Yeah, that's only 12 bottles, and basically it didn't go anywhere.
So $2,400 divided by 12 bottles. That's $200 a bottle.
Yeah. That's pricey.
It is.
I think what they were setting up was to sell it to the government, DARPA, for their soldiers. And of course, DARPA will pay any amount of money to get it, this sort of thing, but it just didn't work. But anyway, so whatever supplies they had left, we got for free.
Interesting.
So we're storing them and then dishing them out accordingly, whatever, depending on needs.
Yeah. So I know for episode 2, right, we're going to be talking about how you isolate ATPSCs and how you use them in clinical trial, essentially.
So that'll be a little shorter than today's introduction. Although I can give you the patient data because we've already published it. So we can go through each one of the diseases that we've treated, the 18 diseases that we've treated, how we did the isolation.
On episode 2?
Yeah, in episode
Yeah.
2.
Yeah, we can. But remember that also then episode 3 and after that, we will be going over also the actual diseases and what you've done. Is that going to kind of overlap?
Well, okay, then what we could do is separate and do the isolation and the basic segregation testing, and then the different formulations that we use depending on which general category of neurological disease or of the diseases, and just stop it there. And then when we got to the individual ones, I can actually show you the patient data that we have
Yeah.
that
Because
we've
then we can
published.
Yeah, because then we can go deep into each specific specialty. So, okay,
Yeah,
perfect.
and a lot of the three we did 3 preclinical trials in animals. We did Parkinson's, myocardial infarct, and pulmonary fibrosis. And I can show you that data. I think I showed you, sent you the figure of that we did for the heart, where we used the genomically labeled. In fact, you can bring that up and show them if you like.
Yep, I'm
Um,
bringing it up right now. Okay, yeah, yep, I have that right here.
I was interfacing with 2 investigators at the time. One was in Columbia, South Carolina, and the other was in Japan. So this is the study, yeah. And we sent the cells to France, and Cécile Duplat at INSERM genomically labeled them with the gene for beta-galactosidase, insect beta-galactosidase, so we could track them. And we could track them either in culture, which is the first panel A, and as an undifferentiated cell, the label was located in the nucleus, those bright green spots. And then as the cell differentiated, it ended up in the cytoplasm. So we could tell whether we still had undifferentiated cells or differentiated cells. And then with respect to the heart, this is the one from Columbia, South Carolina, froze the apex of the heart in liquid nitrogen, causing myocardial infarction. Then we directly injected the clone, and this was SCL40 beta, which is our pluripotent stem cell clone, into the area. And within a few months, in panel or picture B, it regenerated vasculature. In C, it regenerated myocardia. And in C, it regenerated the cardiac skeleton. So 3 different cell types. Now, if you want to repair the heart, you can't just put in cardiac myocytes. Because 1, they need vasculature for nutrients and waste removal. And 2, they need to attach to something, the cardiac skeleton. If not, they just fibrillate in place. But you don't have the concerted effort to push blood out of the heart. That's why we used a stem cell that would form all 3 functions. And that's, shown in this one. My other collaborator was in Japan, and we sent cells there. And what they did was to do transient ligations of the Widowmaker, the left anterior descending coronary artery, to cause myocardial infarction along that pathway. And then they injected the cells through the tail vein. Point being, can we use systemic delivery to repair the area? And they found the same result. They used SCL40 betas, and they repaired the vasculature, repaired the myocardium, and it repaired the cardiac skeleton. Now, they used histochemistry rather than immunocytochemistry, but the results were the same. So that gave us the impetus to say, okay, we can do systemic delivery and still get them to the heart. And then we've done other studies, and I'll send you the pictures, where we did in pig, and we could see the markers for TSCs repairing the heart from the inside out. You're really freaky.
Wow.
And then we used PSCs and TSCs on the outside to heal the heart from the outside in, which is one of those inside out, outside in things. So there are actually 2 forms of that inside out and outside in thing. You just ask for one. The two are with heart, we can use the TSCs through the Tebesian system. They are 0.1 to 2 microns in size. The Tebesian veins vasculature is only 3 to 5 microns in size, which means they're too small for any blood cells to get in. So the only thing that gets in is plasma. It goes back and forth, doing systole and diastole. TSCs can get in, and they basically regenerate the dead myocardium from the inside of the heart, literally inside the heart, and move outward. Then we also gave PSCs and mesenchymal through the coronary vascular system and healed the heart from the outside inward. So they're going in both directions. So we got a quicker repair response than if you just did from outside inward.
Wow.
And this sort of thing. A little different there. And then, okay, let's see. And the guy in Japan, also, the ATPSCs, PSC, SEL40 beta, actually repaired the coronary vessel, the LAD, that was damaged during their transient ligations. You can see the stem cells in the artery cell. I don't have it. I didn't send you the picture of that one. I can do it. I can add stuff to this later, so you can get a more complete picture and whatnot. But it's really interesting what these things will do. And then, I can talk about those later.
Okay.
And we have preclinicals. Did I send you the brain preclinical?
So this is
It would
what
be
I
the
have.
dopaminergic. Let's see, TSCs, embryo. No, you don't have it. We did a Parkinson's model. Okay. And what we did, I'll tell you about it now. I'll tell you about it again when we do the clinical trials. But we created a Parkinson's model by injecting stereotactic. So we had a through XYZ axis, put the needle in through the cerebral cortex into the midbrain and delivered a neurotoxin called 6-hydroxydopamine. And it kills dopaminergic neurons and it wipes out the neural networks. And we got, pictures to prove. And then we waited 6 weeks and re-injected the same area with either saline or SEL40 betas. 6 weeks after that, harvested and looked. In the buffer injected animals, scar tissue. And we could see a glial scar along the needle track. And we use methylene green, which stains glial cells, resident glial cells. And you can see the scar. For the SEL40 betas, we stain for tyrosine hydroxylase, which is the precursor for dopamine. And the cells lit up along the track. So they would be dopaminergic neurons. But we also regenerated the neural networks, which is very interesting. Because most people say, well, I regenerated cells, and they don't talk about neural networks. Well, you need connections, guys. This sort of thing. And also, which we found interesting, which I had not anticipated, is the excess cells that weren't converted in the midbrain migrated back along the needle track and repaired all the damage through the cerebral cortex. So they repaired or regenerated pyramidal neurons, which are specific for cerebral cortex, interneurons, glial cells, and capillaries. So, these cells are very powerful. Not just what you want, but other things as well. This sort of thing. So that brought up the impetus to do Parkinson's disease and human clinical trials. And then we've also expanded to others, which we can get into.
Interesting.
And then pulmonary fibrosis. We created the defect with busulfan, which is a chemotherapeutic agent used to treat ovarian cancer. Now, the impetus for this study was by a OB-GYN resident in my lab called, Dr. Christina Hawkins. And she had friends that had ovarian cancer where they had to take out the ovaries and do actually full hysterectomies. So they lost the ability to have kids. So the question was, can we recreate gametes from totipotent stem cells? So we started with males first because they're easier. They also have a different embryogenesis. And we took interstitial cell, whatever. We isolated cells from a different source and then co-cultured them with conditioned medium for Sertoli cells, which are the nurse cells for spermatogonia. And lo and behold, those cells form spermatogonia. And that's why I can say we can do gametes and stuff in the flowchart. The next step was to implant these cells into, do IVF, in vitro fertilization with these labeled cells and see if they would end up in the progeny. She left to go take a fellowship at Duke, and so the project kind of died right there. But it's open for anybody that wants to try. I'm happy to push it on, see what happens. But, things like that. It's like, okay, you have a very plastic cell population, has unlimited proliferation potential. You can ad nauseum up the wazoo, TSCs will do 3 orders of magnitude every 10 days. So if you start with 1 million cells, you have 1 billion cells in 10 days. 10 more days, you have 1 trillion cells. 10 more days, you have quadrillion cells. 10 more days, you have, whatever. And I've had people say, well, do you have problems with too few numbers? I said, that's not my problem. My problem is too many cells. What am I going to do with them all? This sort of thing. So anyway, but unlimited proliferation potential as long as they stay undifferentiated.
Yeah.
But you can, get them wherever, like some people say, well, we grow up T cell, they do CAR T therapy. They isolate T cells, do the little thing, then grow up these cells, and they have problems with few numbers. And I said, well, what I would do actually is do your CAR T, isolate PSCs from the patient, do your CAR T on those cells, grow them up ad nauseum, and then convert them to C cells, and then do your treatment. And you can freeze down your PSCs, no problem. You can get them out anytime you want, all this kind of stuff. Nobody's bitten on that yet, but I'm hoping, this sort of thing, that you could use the ATPSC as delivery vehicles for both CRISPR and CAR T. Now, this sort of thing.
Interesting.
People have asked, well, what do ATP, you asked me, in fact, well, not exactly, but how do ATPSCs work with cancer cells? Well, actually, I don't know. I haven't tried, because there's 2 possibilities. Either they migrate to the cancer cells and become more cancer cells, or they migrate to the cancer cells and kill them. Now, I have another collaborator who's a DVM that has a lot of friends with cancers. So he has been giving them CNSP just to see what would, one lady has had thyroid cancer, treated, it's incurable. Couldn't do anything. So he gave her some CNSP to try. In a year now, the tumor has shrunk. Okay, that's an interesting observation. He asked about observation, this sort of thing. So that's an interesting observation. Another friend had a optic nerve neuroma, which is actually a mass of cells pushing on the optic nerve at the chiasm and basically shutting off the signals for sight. And she was legally blind. She started on CNSP. And now, with corrective lenses, she can drive. So it shrunk.
Wow.
It shrunk the tumor. Yeah. Presumably. I don't know. I mean, this is, we got to test it. Now, if the ATPCs get to the cancer and form new cancer cells, I got a delivery vehicle for chemotherapeutic agents. Put the genes for the chemotherapeutic agents in the ATPSCs, put them in, let them migrate to the cells, incorporate into the cancer, release the chemotherapeutic agent, and you kill the cancer. Directed cell delivery. Anywhere in the body. It doesn't have to be 10 to the 9 cells to be able to see them and treat them. It could be 1 cell.
Yeah.
So that's a possibility. I mean, it's like I tell people, put your imagination cap on your head. Okay, now, we'll form any tissue in the body, proliferate ad nauseum, tightly controlled. What can you do with these cells? Imagine, what are the possibilities? I said, I have my own sci-fi projects, But what do you think you can do? Now, some of my sci-fi projects we've already started on. One is to create pancreatic organoids, islet organoids, to treat type 1 diabetes. So what we do is we take a porcine extracellular matrix, well, pancreas from a pig, decellularize it. So we just have the extracellular matrix, no cells. We put donor islets, in case of our experiments, human islets, in there, encase it. Then we encase that in PSCs, and then encase it in TSCs. Well, the islets we have tested will form inductive factors that turn PSCs into those cells. So instead of having one beta cell, we have beta cells, insulin, alpha cells, glucagon, delta cells, somatostatin, epsilon cells, ghrelin, and polypeptide P cells for polypeptide P. That's all in the islet. So it controls glucose metabolism, not just moving the glucose into the cells that insulin does. This is a whole body type thing, not just one cell type thing. Now, because the human cells will induce the pluripotent stem cells to form new islet cells, it's self-renewing. Well, it's self-renewing. And the PSCs release PSC conditioned PSC exosomes to the TSCs so they can form new PSCs. And the TSCs release exosomes so they can proliferate. So there's a conversion of PSCs to islets, conversion of TSCs to PSCs, and conversion of and multiplication of TSCs. Self-renewing. Now, I've published on the data of, yes, it secretes insulin, it's human insulin, it's not rat insulin after 30 days. I have not published yet because everybody else thinks I'm crazy, which I probably am, that in long-term studies, we lose human insulin and we gain rat insulin. Human cells converted to rat cells into islets. That data is sitting on the shelf, waiting to be published until people understand the system. And like I said, right now, if I try to publish, they're going to call me crazy and burn me in effigy or whatever. I don't, But anyway, here's that data. Now, it's also immunoprotected because we use TSCs and PSCs that don't have any self-recognition molecules. So the outer layer are TSCs, no self-recognition. Next layer in is PSCs, no self-recognition. So they get by the immune system. So we have self-renewing, immunoprotected, pancreatic islet organoids. Now, we are ready to gear up to do experiments in pigs. We streptozotocin, that kills pancreatic islets, pigs, waiting for them to go hyperglycemic, and then we're going to put in the transplants. And I got sick, so we had to shelve that. But, that's another, we've got all the protocols, we know how to do it. It's just moving to that next step, animal studies, and then into people, and this sort of thing. And there are a lot of other ideas I want to try. Freeze drying is another one, so I can get it out to the masses. But once that is done, I can use oodles of totipotent stem cells to do what I want to do, which are the sci-fi projects.
Gotcha.
They are out there as far as sci-fi projects are concerned. But we've already proven that they'll work, and they were published in the 2004 paper. If you just look. Like we identified SIF, scar inhibitory factor. We partially purified it. I want to complete the purification process, get the gene sequence, stick it into mesenchymal as a production entity, because you have log phase growth and you have lag phase growth. Log phase is your proliferating cells. Lag phases, they just sit there, but they churn out proteins. So you put in you put in the genes for SIF, put them in light phase growth, and collect the SIF, pure form. And then we could use
Mind blown.
SIF on sutures. So when people suture, you don't get scar instead of a scar. You could do it for all types of wound healing without scar tissue formation, all sorts of things. Shuts down scar formation, allows normal fibrogenesis to occur. That's also in the 2004 paper when we did skeletal muscle regeneration. What we did, okay, there are 3 types of skeletal muscle studies. One is you cut a chunk of muscle, but it still has an attacked vasculature, stick it back in where you cut it out. Another one, you pull it out, macerate the daylights out of it, and then stick it back in. The third one is you take a chunk of muscle and toss it. In the one that the vasculature is still intact, you get a little bit of regeneration, but not too much. In the one where you have macerated the tissue, you get intermediate scar tissue formation, but no functional tissue. In the one where you toss it, you have massive scar formation in the area. It just completely covers that area. So what we did was to take the worst case scenario, which is what I try to do, worst case scenario, and give them SKMMP and SIF in combination. And we did it in polycarboxyphenoxypropaine. That was my favorite delivery vehicle because extended release, all sorts of neat stuff. And lo and behold, it formed new new skeletal muscle fascicles that had intact neurovascular bundles, also lymphatics too. So nerve, artery, vein, lymphatic. The blood vessels had blood in them. So it was connected to the, all this. And that's in the 2004 paper.
Okay.
So in combination, you could put stem cells in with SIF. And even if it's in the presence of TGF-beta and basic F, it doesn't matter because SIF shuts down and you have normal regeneration to it. So there are all sorts of things you can do with these items and this sort of thing. It's just how you put them together, what you try, understand what you want to do. Pick out what is available to do it with. Do you need stem cells? Yes, no. Do you need, you need some type of biological agent? Yes, no. Do you need building blocks? Yes, no. Put whatever you need together, go for it. And usually you have a good positive response.
Awesome. Yeah. Amazing. Well, Dr. Young, I appreciate your time, your knowledge, your experience. There's a lot. As you were talking, I was taking notes. I was looking up some terms that I was not familiar with as well.
That's all right.
So I was, because I have multiple screens here, but I appreciate it. I'm actually looking forward to the second episode to where you're gonna, go over. And now I understand more. So I'll come up with like some better questions too as well. And
okay, now I want them in doc format instead of PDF, so I can put it on my computer and just answer that, or the Google Doc file or whatever you want to call it.
Yeah.
Instead of retyping everything.
No, please. Yeah. Just call me and I'll have it ready for you.
Okay. But, I can talk for a millennia on what I do and what I've done.
No, I'm sure. We've got almost 4 hours now, which is amazing.
Yeah.
I've done usually I go long, like 2 hours, 3 hours I usually do. Depends on the guest, right? But I know there's a lot more that we want to go over, but we will save it for episode 2 and 3 and all that stuff as well.
Okay. Sounds good.
Well, you have a beautiful day. And thank you so much again for your time. And I hope the people watching this, even on YouTube and, for future, you guys found a lot of value in this. Thank you, Dr.
Well, if they have any questions, all they have to do is ask.
Yes. Well, you guys have a beautiful day. Talk to you soon.
You too. Thank you very much.